Only 3 or 4 hours from the west coast. 4 hours now, yeah. Right.
And we're live, Dave.
All right, good evening. Dave Butterfield from the University of Washington here. University of Washington and NOAA Pacific Marine Environmental Lab in Seattle. You're looking at some broken pillow basalt lavas in a pillow basalt lava mound on the seafloor at the Mid-Atlantic Ridge at 25 degrees north at a site that we're calling Cluster 2 or Grappe Deux, as we've been saying. This is a non-transform offset where there are some periddotites exposed, and we've been working here for a few days. We found a hydrothermal vent, 325 degree hot smoker at right near where we are right now. We're going to be visiting that vent and taking a lot of chemistry samples and biology samples so that we can get some details on those things. It's an interesting site. It's got lots of shrimp and gastropods and various other small vent fauna that you'll see. And the chemistry seems to be quite interesting also. We've seen some very high hydrogen content, and we just want to get some replicate samples to make sure that we're getting the right results. So right now, checking our nav, we are about 70 meters away from the vent, and this actually looks like a good spot for grabbing rock samples, so we can keep that in mind. In fact, if you see a spot where we can just go right in and grab one right now, we could start right off with that. This is clearly a pillow basalt mound. I'm not sure if they're going to be here or not, but we can log a rock for them. If one of our geologists is listening, you can come in. You don't have to, but we're going to collect a rock sample here since we're looking right at a nice exposure of pillow basalts. One of the things on our list today is to collect the rock samples from right around where this vent is to understand what type of rocks the fluids might be reacting with. What we see in front of us is lavas. Okay. Jeff can log a rock sample.
Is there any good ones that you can grab off of?
Well, we're going to have a look. We might have to see if we can break a piece off. So these are lavas that were erupted on the seafloor in the form of pillows, extruded like toothpaste from a tube, and the surface freezes as it comes into contact with the cold seawater. There's a whole series of volcanic, constructional volcanic features on the slope that we're working on. The slope is unstable and is breaking up into large pieces and sliding down towards the Rift Valley. We believe that as that's happening, there are locally right here some mantle rocks that are normally quite deep that are being exposed. There's a cliff or a scarp that's about 500 meters from here that has periddotites that have been sampled from it. And it looks like we can certainly grab some of these.
Over here, there were like this size, like
So there's a bit of sediment on these. We just want a regular old basalt. That's what we want.
you want us to ask him to make it not grayscale?
I would.
Try to make it not grayscale.
Is it something on
It's not usually
this?
grayscale. No, it's not. Here, I can try and do something.
I can look at this one for now.
Yeah, that's
Great.
That is kind of weird.
Definitely a different color. Always. Like if you compare these two, see that they are adjusted.
Yeah, but that's like fully grayscale. That is fully grayscale.
We are in an economic configuration.
There's a couple of chunks there that could probably work. If we could get one that has a glassy surface, that would be great. It's going to be a little hard to tell looking at these. They've got a bunch of sediment on it. That could be... Actually, the darker one above that looks like it might have some glass. This one might too if you flip it over.
I'll go after the sample. I have a other laptop. Is this an old bin? See lots of. We're at the bottom. We're at the bottom.
There are some tubes there with... What are those are not the same kind of... They're the same species?
Yeah.
And what is it called?
Those are catopterids. It's the catopteridae is
Okay.
the family. Yeah.
Okay.
We collect one last dive.
Okay.
And we will be able to sequence it. So
Okay,
we'll
cool.
probably be able to be more accurate with the identification soon.
Yeah, that dark one would be good.
Look at that bio rock.
Oh yeah, got a little bit of got a bio on the rock. 329.
Get it when it gets in. Oh, okay.
Where would you like it?
Let's see. Let's put it in a rock box.
One?
Rock box 1A would be fine. Oh, wait a minute. That's where that is. Okay.
Yeah, it looks like, I mean, it looks low.
I got a photo, please.
Dave, is this the same event as last time, or is it a new area? Someone in the chat wants to know.
We're going to be going to the same event we were at 2 days ago. It's the... Could we turn that over to see the backside? No, I don't see glass, but okay. Well, let's put it in there anyway.
Yeah, you got it.
Yeah.
But if you can find one that has glass, that's much more useful.
Yeah. Okay. Let's look a little more, see if we could tell if there's one here that has any glass on it. I mean, those look like they're manganese coated. They're looking pretty old and manganese coated there, don't you think? That could be glass
But
underneath,
that
but
round one down there that looks like it has a glassy surface, it's trying to break off.
right here.
Yeah, you think?
Yeah, we could try that. See what it looks like.
Fishing rod or fishing laser?
Could we get the scaling lasers one, please?
Yeah, that one. Try not to squeeze the glass off.
Thank
If
you.
that is glass. What do you think?
Possibly. It might be. It's a very rounded surface. It's so rounded, that's probably a glassy surface. What do you think? It's kind
I'm
of dull
seeing on the
now.
right. That
On the
surface
right
that's on
hand
the
side
right,
of it.
it's trying to swell
Yeah,
off.
yeah. That looks.
Yeah, I
That
think
looks
that's
like
a
glass.
and maybe there's glass on the left too. That's a good one.
Okay, that'll go in with the same one, please.
In the same box?
Yeah.
Thank you.
30. And
I'm not, can you give me a second?
Yeah.
Before you put it in,
Okay.
come hard and fast.
You wanna?
I gotta get-
you want to try to get a description in
I'm
there?
logging
Okay,
the sample
sure.
for the first one with the new keyboard.
You guys good?
Yeah. Two.
Yeah, you can rotate that around for us, maybe. Okay. Yeah, I think it's got some glass on it for sure. All right. Okay, so this one's more rounded. It's got a what looks like partial coating of glass on it, which freezes the composition of the magma or the lava as it's coming out. So it makes it easier to get the composition of the rock. Okay, 2 rocks into rock box 1A. Pardon me?
Keep going.
Okay.
You're running the show.
Are we okay with that?
I'm gonna log it.
So that's logging in 2 samples or 1 sample? 2 samples?
Those are 2 rocks, 2 samples.
Okay. GO1 and GO2.
Yes.
All right. And where is our best depth display now?
37, 42, 37, 37.
Okay.
Okay, we ready to head down the line, or?
Yes. Ready to head for the vent.
Ready, Charlie.
Okay. Just wanted to take that chance to grab
Hey, Jason, that's an updated
volcanic rock while we were looking at a good exposure of them.
Yeah, copy that. Thanks.
Main goal for this dive is to collect a lot of hydrothermal fluid samples for chemistry and for microbiology. Get some vent fauna samples. We're looking to get some of the black gastropods that we saw the last dive, but were unable to capture. Try to get some of those with the suction sampler. This is the only hydrothermal vent that we've seen in this area. There are several inactive hydrothermal vent sites that are nearby. And there are plumes in the area. which is what led us to look in this general vicinity for venting. We found the vent by a combination of looking at hydrothermal plume signals in the water column and using the AUV to identify structures on the seafloor that could be vents. Seen some shells on the seafloor, possibly some what are we looking at down there? Can we get closer to that to see what those are on the bottom?
But just in terms
Oh, those are last one. Let's see, these are last one. Sure. That one, so. Yeah, it's the same as this column. Okay.
Looking at a slope with
Those are the same gastropods.
okay, there's a whole field of these snails, gastropods.
Actually, it is a much more common location.
This you were saying they're mostly found on the periphery of vent sites?
Yeah, this is where I would expect to find them usually, not that close as the last time. Yeah, snails are different varieties.
You're looking at the live stream from ROV SuBastian from the Mid-Atlantic Ridge from the Schmidt Ocean Institute vessel Falkor 2. Large congregation of gastropods, snails. There were a lot of these seen at the vent site as well. Current depth says 3758.
Can we zoom out a bit just to have an idea how extended is this snail field?
We don't see any active venting right here, but there are obviously a lot of snails. Looks like some possibly sulfide type sediment coming down the slope. Lots of tubes. The rocks we see there do not look like pillow basalts as far as I can tell. They're big and blocky. Possibly very broken up pillows. That's a very large field of snails.
just a few.
All right, this depth is maybe 20 some meters below the depth of the vent. If our depth is good. Okay, let's continue up.
It's a pretty large area.
Oh, wow. Pretty rat tails, pretty grenadiers.
Pretty rat tail fish hanging out in the water column. Question, do the snails have a blue foot? To me, they've looked mostly white. I've seen snails elsewhere that are very blue. Let me ask the biologists if they noticed any blue on the samples that they got. Are they?
Any blue?
Yeah, is there any blue color to their flesh? No, they're all
No.
white.
Yeah.
Nope, they are all white.
There's actually more of a cream color, but I think it comes off as really white down here.
Okay.
If we want to be really picky about it.
In the lab, they're somewhat greenish looking. Looking like pillow basalts again. So we've been out here for, how many days now? Let's see, this is the 29th. We left 26 days ago, and we're down to our last week. There you can see the sulfide chimney coming up. This portion inactive at a depth of 3,758 meters. The fish is swimming upside down. Interesting.
Yeah.
Doesn't care which way is up, I guess. See the squat lobsters on the blocks of sulfide down below, and more snails scattered around on the ground, and some anemones. This is probably an inactive portion that's below the active chimneys. So as I was saying, we found this vent site by a combination of looking at the hydrothermal plumes with CTD tows and CTD vertical casts, and looking at the AUV bathymetry and the water column sensor data from the AUVs. There's an eel. That's an eel. It's not. Do you guys know? Have you seen that one before?
These eels,
Yeah. Yeah. Pretty common.
it's difficult to say these, but
Yeah.
they look pretty similar. What we didn't saw is this huge extension of the snails. That is something very unique, I feel. At least in the lab.
The lasers are 10 centimeters apart. You can use them for estimating the size of things. Some old sulfide chimneys here. We were seeing the greenish colored, orange greenish colored sediment coming from this a little bit further down where we first saw the snails.
Yeah, yeah.
These are Ketopterid. Say Ketopterids?
Yeah, so you can see how many of them are like coming out of their tubes.
Yeah.
The ones that are feeding from the bulbs, they are filtering from the environment probably now.
Ketopterids in tubes, filter feeders. Commonly live in sediments.
First time I've succeeded.
Yeah.
Oh, there's some active fluid flow there. Some smoke coming up. And a swarm of shrimp. So this is a deeper portion than we were looking at the first time at 3,750 meters.
So those look like shrimp eggs. We need to zoom
Faint black smoke coming out of with the fluids here. Shrimp are swarming around right in that fluid. Quite a thick mass of them. Looks like lots of dead shrimp around the edges.
It really seems like a
And
dying smoker. But the majority of the shrimp are dead.
there's a lot of dead shrimp right here. Yeah,
It seems
and they're
like the
coated.
activity has gone down and they have been left without fluid for the bacteria and probably died. I would say, I don't know. It's,
There's plenty of venting right above though.
yeah, but
And there's venting right there with the black smoke coming
it's a
out.
very small orifice.
Yeah.
I'm not sure it could
And this
sustain such a big mass of shrimp.
and are the snails scavengers eating the dead shrimp?
Yeah, probably those gastropods are considered scavengers. So
Yeah.
yeah,
Well, maybe the fruit is not reaching up.
yeah.
We were just saying the ones that are alive and moving, they seem to be moving very slowly.
Yeah, I guess they are dying.
Yeah.
They do not have either enough energy or that was very active and now it's not that much. Something like that.
But still, then, I'm guessing
They-
it's the quality of the fruit that
But
what.
if you check the laser, this is 10 centimeters. So what you're seeing is this.
Yeah, but that's like, remember other places like at time or so, you can have very little holes and all
That's
the still.
Can we move up a little, just a little bit to where there were more shrimp that were swarming around a little?
And the ones that are above, they seem.
Okay, that's
another
fine.
species.
Yeah.
You will see that the juveniles.
So there's a bare spot there with some glittering sulfide minerals in it. Mostly clear fluid, a little bit of black to it. The shrimp are sort of dark gray from living in that water. We're talking about why there are so many dead shrimp right here, and potentially they live by chemosynthetic symbiosis and generally require hydrogen sulfide as an energy source for sulfide oxidation by their symbiotic bacteria. So potentially this water doesn't have enough sulfide to keep them healthy. You would think they would have the ability to know if there's sulfide in the water or not. I mean, We obviously smell it. They must have sensing for that.
I mean,
Uh,
my hypothesis would be that that used to be more active and enough to sustain all these guys here, and probably either the chemistry change or the fluid start to venting less, and then
Yeah.
and then they die. That's what I would
67 was only a little bit.
imagine.
We could take a water sample, but I want to save those for getting our high temperature water first. So if we have any left, we'll come down here and see what's in this water, so you can test that idea that they don't
I
have
mean,
the.
if those here are healthy, that means that there is sulfite here at least. So
Yeah. Okay.
here do not
We
have
can
problem.
continue to move up.
That's great.
So
Let me see here, 180.
I would expect there's at least some sulfite in there because the black smoke particles indicate metal sulfite high. I think higher than we've seen anywhere.
So here.
And the ratio of hydrogen to methane or hydrogen to hydrogen sulfide is quite high.
I mean, they might
So
be-
hydrogen here is certainly the biggest chemical energy source. Some black smoke. Let's have a close look at the black smoke there.
we'll have to look for the last one.
This is a potential site for fluid sampling. There's the vent fish in there.
yeah. It's just the
Okay, this smoker at 3,747 meters is a possibility. This is the lower portion of the high temperature vent. We did not sample down here the first time. Looking for a second spot to sample. We're going to resample the place that we went to the first time and one other spot. $37.47 at heading of 52.
Did you make a marker today?
Yeah, sure. Yes, it's good work. What do you think of that as a sampling spot? Seems like it should be doable, yeah?
Yeah.
Nothing on the backside to get in the way? I think I want to look a little more. Oh, lower smoker. I don't know. You have to have a name for it?
Yeah, it's gotta be good.
Lower smoke.
Blowing smoke.
Blowing shrimp.
If you got to have a catchy name, we'll call it blowing smoke.
Smoke in the wind.
Little bit of black, diffuse black smoker there. The smoke from this stuff is extremely black. The samples we got up, the water samples we got up were black in color. Even the ones that we filtered in place came up black looking from continued precipitation of what could be pyrrhotite, possibly. Currently just getting a good video overview of the structures here. Quite a lot of smoke coming out of that one. The spot that we sampled the other day was 3,734 meters up around the other side of this structure. It's growing on a steep slope. Let's keep going up and around to the right and look at this from the side if you can.
Coming around to the right.
Yeah, how high is this smoke?
The spot we were at before was 3734 and we're at 3747 right now.
Yeah, we would be wrapped around it then.
Can we come around to the left and around that side?
Yeah.
Okay.
Just to double check, are the lasers on?
They've been on. I
Oh,
think
yeah,
they're
they are on. They look like so faint.
Oh.
Yeah.
okay. Very smoky over on this side. Shrimp in there. Let's see. Looks like another potential sampling spot for diffuse fluids. Wow, that's a tall one there in front of us.
You want to mark it?
Sure. 374.
Something smoking my left, so you really keep some distance. Yeah, that one.
All right. Not going to see much on this side.
We're in some serious smoke.
Yeah. Okay. Tell you what. Why don't we back off, go back down to that base smoker, and we'll sample there, because I don't think we're going to find a better spot to set up. We have an AUV in the water at the same time as we're using the ROV here. That'll probably be the last AUV mapping run that we do in this area. And we're also planning to do another ROV dive after this one. This dive is primarily sampling at this sulfide structure at the smokers. And the next one is going to be a geological transect to try to figure out where there are mantle rocks here and what layers they're associated with, possibly to get some evidence that there may be mantle rocks directly under this site that are covered up by a layer of volcanics. Yeah, I want to set up the sample here.
And you Butterfield in here, but why not more?
I think we'll be setting up first to try the high temperature probe, ear probe, and possibly try to take a chunk of this sulfide chimney to collect. The AUV is off mapping to the south of us and deeper. AUV dives last for deck to deck about 18 hours. And it's been in the water for about 5 hours now. So there's around 13 hours left before it comes to the surface. And we need to be up before then, and we need to leave the bottom in time to get to the surface. So we've got about maybe 8 hours of bottom time.
Try rotating it a little bit.
Just getting set up to try taking a temperature measurement here.
I'm gonna try and do it a little bit now. Make sure I'm stable.
Going to do several things. Temperature measurement, try to grab a piece of sulfide, and then once we've got that and we see potentially have a clear orifice to try to sample, we'll get the hydrothermal fluid sampler up and take a series of samples with that. Well, we can zoom after we get settled in. What did you want to look at?
It seems that there is
We
something
we
with
saw
the.
some, it looked like we found some polychaetes in the black smoke, which is crazy.
Here, basically. So I don't know what to add. I don't know if it's something real or not. Ah,
Yeah, like right where the lasers are.
I see here. This particular is. It seems that there are the gastropods, the black gastropods, and probably something else on it. Wow. Here. Just where the lasers are now. Yeah.
Excuse me. It definitely gets messy when you're trying to sample something like this. The flow is coming out from a fairly diffuse structure. It's probably very breakable. It'll probably fall apart when we touch it. We will see if we can get a temperature measurement, which will tell us how hard it is from poking around with a temperature probe. Those are our titanium gas tight bottles on the front of the sub. They've got some polypro line handles on, polypro lines that hold them in place. Bungee cords there, keep them from falling off the basket. This would not be a good place to drop one. It's very steep.
Yeah. You push right to the left and rotate it.
Do we have our temperature display anywhere yet? I guess we will.
Which page is that on?
There we go.
Just watch the flow there.
Getting a little bit of warm water there. Just starting to move around to try to get it in the flow.
Small things covering it up. Those ones that
Up to about 70 degrees right now.
yeah. Gotta keep it as close as do they have any essential oils? The two spots on the back.
They can detect very special items with hydrothermal vents for the reactions.
Okay.
What do you want? What do you want in return?
Cup of water? No, nothing.
I think I need to drink. There it says HT probe 1058.
58.
Trying to find the right spot here. It's a lot of smoke. Can't see where it is.
50, 60, 70.
That's yeah, over there. 449 HTC.
We're expecting this is in the 300 plus degree range.
Want to reposition?
If you want, we can try taking a sample of it, which will probably lower that opening down some.
Meters 80. See the last one.
80, 85, 90, 100.
Okay, we get above 100.
Yeah.
10.
Funny, it clears up in one spot. You can't see it.
Why don't we try sampling some chimney? We can sort of break it down and see where the opening is.
Okay.
Yeah. Do you guys want to say something about the snails?
Oh,
Or the shrimp that you sampled? There's questions about
Yeah,
it.
the question, yeah, I just saw Jesse's question. So, we did end up having a look, I think here. We
Okay.
did end up having a look at the little kind of pyramid shaped capsules that we thought had been egg masses. And we ended up looking at them under a microscope yesterday and puncturing them, and it was in fact all of these
What happened?
thousands of eggs fell out, so they were in
Just the
fact
cable
egg
went down?
masses.
Sorry, we dropped the temperature, bro.
Oh no!
It's attached. Okay, sorry, I missed the end of your story about the egg potential.
Just that
egg.
it was really exciting that we figured they were egg masses, just given the large numbers that we're seeing of them all congregating in one spot. But then to actually look at the little like pyramid shaped gelatinous masses under the microscope, and for each one of those little pyramids, there's this egg sac. And then to actually puncture it, and all of these, hundreds and hundreds of eggs came streaming out. So we know for sure at this point that they are brooding.
Yeah.
But we know they are eggs.
Well, yes, we know that they're eggs.
We still need
We
to
strongly assume that they're the eggs of the gastropods, but
Okay.
we are doing. Well, Sean is doing genetics to confirm.
All right.
No, it's not that. It's just, we have this bit here.
So they have the egg masses, and they have the gastropods, and they're going to sequence the DNA of both of those to see if they are the same thing. Presumably they are. And many snails were congregating in that area, appeared to be putting egg sacs, sticking them on the sulfide chimneys.
I think we have a little bit more chinchilla. Is that not too, that might be too close to us.
Might have a little more fortune than you gave it a reach down.
Take
water? Want to go get a coffee?
Sure. I could just use water in this since I cannot find my water bottle.
Okay.
All right, thank you.
Porch out.
Yeah, porch out.
Just watch those,
Okay, just retrieving the temperature probe here. So we didn't finish getting a temperature measurement because it was too difficult to see where the opening was. So we decided to take a sample of the sulfide chimney, probably open up a more visible opening to measure and to take water samples from. And just retrieving the temperature probe now.
Let's put it in time for the next time.
All right. Next, we're going to use that arm to try to grab a chimney piece. Do you need to move that or what?
Sorry.
We're going to try to grab a chimney.
Oh, okay, sorry, I thought you wanted
Piece.
to, yeah.
That'll knock it down and see if we find an opening that we can measure.
Yes, we can't push the arm right into the hot stuff there, but if you see a piece on the outside.
Okay. Because you'll melt the plastic or what, the
Because
fingers?
it seals in the arms.
Oh, it seals in there. Okay. Do you think you could?
Yeah.
Yeah.
So, you want that little piece kind of sticking out right there?
I want part of it where all that smoke was coming out that you were trying to measure. Open it up. Will that be
Just gotta be
bad for
really
the
careful
arm?
with- Yeah, just gotta be a little careful.
Okay. Thank you. The setup for all of these things is the hard part. Once we get in there, we'll be taking a lot of samples without moving. It's just getting to the point of getting a good temperature measurement and finding where to put the intake nozzle.
You may not be able to get a good sample if we can't see it.
Yeah. We're up above it a bit right now, right? Go back down a couple meters. There's so much smoke coming out of here that working down at the base is easier than working higher up because they're totally blinded by all the black smoke above.
I think this is the lower location. The vent we were just at was it not?
We are a little deeper now. Yeah, we could try this
At
one.
least we can see. Yeah, this one's much better for the see it.
Not as much fluid coming out.
Yeah, so what do you want to do?
Did we? Which way did we move from that one?
We just went down
Is it-
basically.
so it's straight above us.
Essentially. At least we can see now. We can walk back up.
Yeah, let's try to go up and look at it again. Yeah, our heading was 52 the first time we were looking at it.
So much smoke there.
That's probably the smoke from the vent that we were at, right?
Probably.
No.
Looks like it's blowing back towards it, so let me give it a bit of time and it may clear up. Or if in the meantime you want to go back and look at that one, we can.
Well, this little one is a possibility, but there's a lot less water coming out here, so I'd like to try to get that bigger one.
What was the depth of it, Dave?
It was 37, and our heading was 52. I think it's the one off to the left. I could be wrong.
Yeah, that's the problem. I see that.
Yeah, we may have disturbed the flow of it. Yeah, the temperature broke it.
Can we just drop down a little and go to the left? That one could work if we can't get into this big one.
This is where it's coming from.
Yeah, this is the one we were looking at, and we were, I think, looking at the left. Oh, maybe it's not.
This is a different
Yeah.
one, Dave, 'cause
Is
I don't
it?
have any snail trail on it.
Okay.
Seems that it's a much larger complex than what we thought in the first time.
Yeah, it extends down the slope quite a bit. It's at least like 15 meters vertical extent up and down the slope, maybe more. Oh, well, here's a lot of choices. Mostly diffuse.
Oh, did that this one right there? You want to try to do this one, Dave?
Yeah, let's have a look at this one.
That much better it is.
Yeah. Huh. Did we really move that far over? That's pretty.
Yeah, we went over about
Okay.
15 meters.
Okay. It's a big smoker complex here.
This kind of cluster of. See, here. I'm already distinguishing the white stuff here, look which ones.
Well, originally it was, but it's round, so that felt like it could be in my face. This is flatter. I'm gonna try that, but that thing's gonna be right in the camera.
Let's try it. You want to take a temperature probe first?
Yeah, let's try that. These look like they might be a bit difficult to get because they're really diffuse. They're probably going to crumble, but we'll see how much of the opening we have after they fall apart.
Yep, you went right back to the right pose. As soon as you got that look down. You actually look like you're interested in my knowledge.
So it's care about. Has a beautiful side.
Stunning.
Is that too high? Well, we'll grab it real quick.
Looks like your bumper is kind of underneath it, huh?
Watch out.
Strange.
Yeah. That's
If you want, we can start with the fluid sampler nozzle.
Yeah.
If that's easier, you can knock things down with it.
Yeah.
Yeah, do you want to try it?
Yeah, let's try that.
Yeah.
Yeah.
No.
That's great.
So complicated.
Okay, we've got our titanium nozzle for the fluid sampler out. You can see it's already coated with some black smoke from the previous time we used it. We'll see how soft these are.
Are you able to comment on your Microsoft? Can I keep this out? I'll just gonna push it on the left side.
Yeah.
I think so.
There's your dark snails.
Yeah.
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 Oh, see here, that one. See the polychaete on the top? Yeah. You see the temperature variation there?
Mm,
Maybe that's just camera
stand
coming over
by.
Can you just set it down? Just a piece of
Yeah. Yeah.
it really seems,
Not much yet. No.
Okay. Let's zoom out one more. Let's see.
Yeah, there you go.
That's really going to be hard to get that. That's
Yeah,
the way
it's
it's.
all over the rock.
Yeah.
Look up left, I think there's another one.
The one that's sticking out.
Oh, yeah.
I mean,
Yeah.
you could try going in there. You're gonna have to get right down to the base of that thing. It's too small and crumbly.
What if I can knock it off?
Yeah, I would knock it off down at the base and see what we have. It doesn't look like a whole lot of water coming out of there. Yeah, that looks a little too weak to sample. Given how.
2 weeks?
Yeah.
Is there one here in the screen that you want to have a look for?
Mm. What's the one that's sticking out? How high does that go up? What does that look like?
Give me to knock that right off there.
Well, can't see where it's coming out really. Let me look over there, if you don't mind.
It's a two.
Yeah, I don't do it.
Uh, I mean, you can see the one on the left, how it's just
Yeah.
big and hollow, but there's very little coming out.
See if we can
These are really just, yeah, see how it's really soft.
Seems more gray than black.
Yeah. There was, maybe that little one that you saw that was all by itself, just to the right of this, just a couple meters away. You remember the little one we saw coming into this?
So, yeah,
It just,
come around.
just to the right. Yeah, I think it was just by itself, and it had black smoke, a single little spire. This whole section here looks like it's just really diffusing, which is hard to sample.
Even that one there?
It was a bit to the right of this. Keep going around to the right at the base here. Keep going to the right. It was a bit separated from this group. Yeah, maybe that. Let's try that one up there. Yeah, that little one right there. This one's a little more manageable. Maybe we could find a good spot in there. And do you think you could sample the top of that, or is it too hot to?
We
Grab
could
a hold
probably
of.
knock it onto the porch,
Okay.
something like that.
It's kind of a question for Melissa, I suppose. We'd have to knock it like maybe into the bio box.
Yeah. Deal with HFS before we put it away and then get back out.
Okay, you wanna do an HFS here, Sid? Or you wanna sample the top first?
Let's see what smoke is coming out the top of there. Well, I'd kind of like to try to take a sample of the chimney.
Okay, so we'll go back to the HFS. What do you want?
I guess you got to use that arm to do it. Yeah.
Use the probe,
You could use the probe to knock it over. Yeah.
But if
Yeah,
you want to
push
push
it.
it just falls.
Yeah. Well,
Yeah, so like,
open the bio box and.
the thing is, I can't get my jaw on it, so the only thing I can really do is knock it towards us. So
Yeah.
what we could do is open the bio box and fly up and try to knock the top into the bio box.
Yeah.
Very delicately, so
burn the arm.
Right. Well, you can use the nozzle for that.
Yeah, we can use that. That would be better for me.
Yeah.
Let's do it. And then into BioBox 1 is okay?
Yeah, the BioBox on the. I mean, you need to use the small BioBox on the corner, right? I mean, you can't knock it into the one in the back.
Well, we can knock it over everything and just smash it. You know, we've done this before. It's not
Yeah.
been pretty, but
Yeah,
but yeah, that's and there's nothing in there right now, so
right.
that's the better one.
Okay, opening up the BioBox. We're going to try to get up next to that little smoker and knock some of the chimney into the box as a sample. We've not been able to measure the temperature yet. Getting ready to do that.
That's a good idea. Let's say we use it. I think it's up. I think it's up. Up. Enough. Maybe not for now. I think you're right.
I honestly do not have an idea
Kind of want to chill for a minute and see if this clears out.
Yeah. Excited. We saw a baby right out of
Want to put in a marker spot on this one?
Yeah. So,
These are the ones that were sampled last time, so probably one of them is the one that fell off.
Okay. Is it not? You mean those are suspect?
Yeah,
Okay.
they're all suspect to be enclosed.
Okay.
So that's why we're thinking 3 and 5 over the hood, and then the rest of them just while you're sitting in diffuse area.
Okay.
6 being like the primary one. Hopefully it works. But sort
Okay.
of like, I'm not Schrodinger sample. Whether these will
See the best,
be
just
close to
close waiting for the right one.
Okay. Let me try to put a note on here for that. Let's put that right on here.
Yeah, it's clearing up, I'm gonna create that on it.
Basically, once you get it against that round box, I'll come back to it.
It's fully wrapped out.
Okay, we've got our bio box open and gonna move forward to try to knock the top of this chimney right into the box.
Yeah, I'm against it.
Gotta get closer. Yeah.
Something fell.
We're against it.
Okay,
Pretty dusty.
Okay.
let's have a look in the box. It looks like some of it went into the right side of the bio box, which would be 1B, although might have gone into 1A as well. And some of it probably went into rock box 1A.
It was pretty,
It was dusty.
Yeah, there's a chunk of it sitting on top there. Just knock that in.
Did we get some?
We got some, yeah.
So bio box.
I don't know if we can
1A,
see.
1B, possibly into the.
Some of it went between.
Where do you think most of it?
I think most of it went into 1B.
Okay. Geo 3.
Bio box
do you
1B.
want to try another one?
It's right there.
You can, yeah, you can try to knock some more into
You're gonna
that.
get some in A and B. It's not. It's very crumbling.
Yeah, okay. Yeah, go for some more.
It's gonna get messed up.
Yep. You can see some sparkly sulfides coming out of 1B there.
I'm against it there. Awful low, but
We're not wanting to put the arm right into that because it's got some plastic parts that will melt, but the fluid sampler nozzle makes a good tool to knock these things over. Whoa, that's a big one. All right,
All right. So where's that?
it's all over the basket.
Ta-da!
Uh-oh. Don't fall away now. Okay.
coming up.
You can stay down by the vent if possible, or don't. It's still hot. We've got hot water coming out of it. All right, that's a very big sample.
Maybe right science come in.
I think Melissa will really like that. Although it's a lot more material than we need to analyze, but it will tell you a bit about the zonation in the chimney. Yeah, you betcha. Let me do that.
Oh, yeah, that's still
Yeah, it's still venting hot water.
Cool. Thank you. You want that gas strip? Yeah. Yeah, you got some gas strips on there that we're looking for.
I mean, I think it's. Well, we can decide later. Is that going to stay put there, or?
Well, we probably should break like one of the edges off. And
Put it in the middle bio box, in the big bio box.
Yeah, we could probably get some of it in the.
The neat ones?
Yeah.
Yeah, that would be great because they don't look bio ones.
That would probably push it to the side of the bio box. Do you want to put the arrow and land out?
Yep, it was totally one.
And then we can do this like kind of do this.
We don't want to knock the whole thing off by opening the box. You might want to just close BioBox, the small one,
Across.
and maybe slide it on top of that.
Yeah.
And a lot of critters in there.
On
This is Geo 04. Is the chimney, big chimney piece is 04 and the fragments are 03? Or do you want?
What? It's all one chimney, but I guess it's going to be in two places. So yeah, 3 was the first one.
Where's rocket?
3 is
I
in
feel like it's
mostly
tougher to
Biobox
rocket on
1.
Right.
This thing is, if it's Geo 4, is laying on
We're gonna. I think we're gonna. We're gonna.
here. Or, well, it's like this.
Yeah.
We're gonna try to. We're gonna try to get it into what's labeled Biobox 3 on the diagram,
Okay.
or Biobox 2 maybe. Sorry.
Well, it's kind of laying on Biobox 2.
It is. Well, if we
Open it,
can.
wouldn't it slide on?
If we can.
You can support the back side.
What do you think?
Okay.
Can we rotate it a little bit and then
You want to get
open
as
the
much of
box?
the piece in the box you can.
Yeah. Try to get the whole thing in the box.
Yep. We're gonna find a nice spot to land out.
Oh, okay. All right.
For your business.
Alright, this in the notes.
Mm-hmm. It's probably not. It's not. But it's hard to find a good spot down here. I think it's. Well, we're gonna sample the original spot too. I'm looking for 2 sites, so this will be.
Okay.
Yeah. We'll just start with this one instead of the original.
Makes sense.
Other people.
Still hot.
There were several here.
Dave, are we trying to collect all the chimney?
Yes.
Okay, perfect.
We're trying to just open get it rotated around and
I think there are some astronauts even under the screens.
okay.
So not only the one that was visible more close to us.
Okay.
So if we get these, we already have some samples.
You got your sample in there, yeah. Okay.
benefit.
Yeah.
2 birds, 1 chimney.
I think you need to rotate this over so we can open it. Start flipping it in. Or I can slowly open it.
I'll support it back. If you come up, I'll just support it, turn like this and then.
So we're just doing it in Biobox 2 at this point?
Yeah, just for now, so we get.
Are you gonna push on the bottom end of it, or? I don't know. Yikes. Okay, keep going. In the box.
Where is it?
Yeah.
We're gonna open the other one as well.
Let's see if we can get it mostly in that one.
Oh, okay. It did, it broke in half.
Good.
Goes.
Turn into a shrimp. Yeah, and then it's got
The shrimp have been probably swarming around that sulfide chimney for quite a while, and it's still warm, so they're still looking at it as a place to get some nutrition.
You can just be first.
I think you gotta
It's literally a POG.
What? Well, it is and it isn't. Like the one piece, I thought I was gonna like knock it off, but that would be like art, and it just disintegrated into 1,000 pieces. And then this one, that was gonna be some.
Or stop.
Dreams are following.
Yeah,
their
here it
hearts.
is. There you go. This one can come up as you're doing it. There you go. Yep, yeah, big one was in there.
I think it broke in half and went in there.
Yeah.
I think we've got more than enough. You can probably get rid of that piece that's on the top there.
Well, it's too bad it didn't come up in one piece, but.
Well, it's quite big, so there's some big pieces in there. Now, is the bio box going to close? Probably not. All right.
Can you close power box? Can you close power box?
We never opened three.
It's cracked open right
Yeah, the one on the left is higher than the lid.
Oh, really?
Close it down, it'll be all right.
Blocks of vent.
Wow. Okay. That's a very large sample. That thing must weigh 70 pounds? I don't know. Still gonna be able to.
Yeah.
So that was Geo 3 and 4.
Geo 3 and 4. Geo 3 is in Firebox 1B. And Geo 4 is in Firebox 2.
That was at 0200.
You mind if I go up and dust off?
No, just don't lose the vent that we just sampled, just took this
no,
from.
I don't have to go dust off. Oh, okay, so you just right up here?
It's, yeah, I think they just came right down, dropped down a few meters. Should be right above us.
Can you pull in a little bit? See what you're looking for?
I'm not sure how far we moved, but we did mark it. But it's,
Oh, yeah, I didn't mark it.
it's probably that right in front of us.
Coming here, and coming faster. Maybe like 45.
Yeah, it's
Right up here.
right on top of that little ridge. So we looked at several sites down here, and they were either all in way too much smoke that we couldn't see anything, or there were some that were very diffuse that we probably couldn't sample very well.
Yeah, which is,
And
yeah,
this-
that's one which time.
Yeah, this one looks like at least we could get probably a good sample here.
So this guy right up here, right?
I think it's we knocked off that piece right there. Yeah.
So like right here?
Yeah, right in that stump there. There's a good bunch of black smoke coming out right there. That's what
Right
we're,
here, sir.
yeah.
Like right around here? Like
Yeah, that right there, and it looks like the little part on the right.
should we move
That's
this
a
closer?
pretty good.
Is that the chimney we shot?
I think so. Yeah.
Okay.
Does it not look right?
Yeah, that looks like it. It's the mark from where we were pushing up on it, right?
Probably, right there.
You're on the HFS range.
Yeah. Well, we'll use your temp probe too, but it could be in either order.
What's that? You go for temp probe first?
Sure. Okay, yeah, temp probe's good. Okay,
I'm ready.
yeah. Okay, now we can see where to put the temp probe. See what kind of temperature we get.
Behaving normal, right? Yeah.
Seem to be all right. Say that again? Yeah, yeah. Real quick, then just shoulder right and touch. Just want to get you off that aspect right there. Just rotate counterclock. There you go. Now, come over. Cover, you're underneath it now. You're starting to go up. Hold it for a second, buddy. 2 seconds will be able to keep on. We'll take care of you in deep in there.
Getting 100 now, going up. 200. 275. 290.
Okay, that's good.
Just gonna get a real good
294. Okay, we're getting 294. It's pretty steady.
You want to move it around a little bit?
Well, you could try it.
Just make it a little bit
294.
Doesn't seem to come.
That's what I got.
Pretty steady around 294 there. Okay, I think you got a good number. 294.
Yeah, 294.
Yep. Okay, I think we can sample that. That's a pretty good opening.
Where you want to put it at then? Is that what it was, or?
What's the temperature last time?
3.
Something, or even more?
I would have to look that one up.
Or it's just
I forget. I don't remember. I thought it was 324, but
Probably. Adam tried it earlier and it like flipped out, so. That's why you ended up putting it on the, over the gas tank. About the impact of the. Schmidt's cable between the creeks might be alright, so they.
307. But that was the HFS. I think the temp probe got 324. So the fluid was a little hotter up on the upper backside of this, on top of the structure. Getting 294 down here. And we're going to use the fluid sampler now.
That's your clear. Torch is coming in. Start on it.
Yeah. Yeah, we want that. If it is, it's certainly. But it looks like it is.
Okay, we've got our pump on. Temperature is starting to go up. Just getting about 9 degrees. Yeah.
Could you explain what this smoke actually is?
Sure. So we've got hot water coming out of this orifice here. You can see it's lined with these sparkly metal sulfides. Could be looking like pyrite in there by the color. Some black coating on the outside. When these hot fluids mix with seawater, they precipitate these minerals, or when they cool off. But the black smoke is primarily what happens when you mix the hot fluids with seawater, and metal sulfides become supersaturated and make these black smoke particles. And the particles are mostly metal sulfides. Here they could be pyrrhotite or sphalerite, which is zinc sulfide. Iron is usually the dominant metal, and making different mineral phases with sulfide when they cool off. Okay, getting black smoke in the back. I'd try moving it around a little bit. We got 155 degrees with that.
It's about normal, right? For your temperature probe versus.
No, it should go up,
More than that?
or at least we'll try.
Bring it a little closer there.
Keep going. I'd go back a little bit, and okay, that's starting to go up. Going up. Going up gradually. 120. Maybe
Be like this.
maybe try moving it to the left a little bit. And it can dig in there a little more if you need to also. $110.
The body fluids.
We need that.
115. Maybe try putting it, pushing it straight in a little bit into the material.
Try to put it straight down, a little bit more straight down.
That's going up 80, 120 going up, 140, 160, 180, and 200. 210.
You're frozen, right? No.
I think that's probably going to be good right there.
Freeze it?
Yes.
220. Okay. Well, we're not getting 300, but we're getting a pretty good steady temperature. I think we'll just do this. Okay, so we're on sample 5 now.
Okay, noted.
Correct.
Okay.
We're starting with
Well, why don't we start with a bag?
523.
Bag 23, we'll try.
Yeah, they seem very associated with this.
B times 3.
So this could be S0498, HFS 05, B23, and we're at 3752 meters. The heading is 111. There's a nice shiny piece of sulfide. Okay, I guess we shall start this one.
I'm ready when
The other day you got 300 mils of gas, right?
Yeah,
And 300 mils of water, more or less.
yeah, that was about it. 295.
So maybe 350. All right, starting that one. Uh, why don't we call this one Broken Spike? I can't tell if that one on the right or not.
Yeah, you do. I can see it.
See something there?
I see the shimmer around it.
Okay. All right. So we're getting a very steady 223 degrees.
Can you see water coming out?
Yes.
Okay, cool.
See a little shimmering water coming out.
Uh-huh.
See plenty of smoke coming out on the flush side. The T2 is up to 62. Okay, this sample's coming along, almost done. Okay, that one's done. Stopping that one. Let's just take another one, 22.
It's a flatworm. Flatworm. Yeah.
Just let me know when they're ready.
Okay. Okay, start that one now. Temperature's up to 233.
That's a low flatworm.
T265. Is there any more light available? We should maybe next time maybe we can point the camera up a little more or something. Or move it.
Better now, see?
Yeah. Yeah, you can see it. Okay.
Yeah, maximum. But it's in others. We are rising the day. 2:42 now.
I slowed down the pump, see if it would go up, and it did go up with slowing down the pump.
Okay.
It's probably also sealing itself off.
We're done, right?
Yes, that one's done. And
Did you see?
now I think the filtered ones are 16, 17, 18.
It was written, right? Put the back, take out the sticker.
I saw the sticker on there, yeah. I didn't take it off, but I know 17 and 18. I believe it's 16, 17, and 18. So I think I'm going to take one of those. We'll just make it 18. Temperature's up to 255.
Okay, I'm ready.
Moving the valve.
We go for 18.
Yeah.
Okay. Same volume.
I guess so.
Okay.
B22.
B18.
We did B22.
B22 was 06. And now we're going for B8. I'm ready. Yeah.
Ready. Okay, starting the next one.
Okay. Vegas.
They want to split that one up.
Hopefully he doesn't run away.
Turn it left. Still see the stream.
No, I don't see it either.
Oh, yeah.
Did you see something happen?
Yeah, it's still shimmering.
All right, I stopped it. Now I'm going to restart it. Let me tell me if you see something happen.
Yeah.
Yeah.
I saw the poof.
Okay. Temperature keeps gradually going up.
I saw some. That's it as well.
We are trying to make sure we get a good set of samples for chemistry on this. So we're taking lots of them.
Still see
Each one takes a few minutes to fill up. We're putting these in collapsible Tedlar plastic bags. Can handle pretty high temperature. And gas pressure up to a certain point. One of the samples we brought back had almost the same amount of gas as it did water when it got to the surface. So we're trying not to overfill these things. Okay, stopping that one. It went over the volume.
Oh, okay,
Hopefully we're all right.
okay. How
Because
much volume did
when I
it?
stopped it and restarted it, it didn't keep track of the count. So we ended up getting 500 mils,
Okay,
505.
505, okay.
Maximum was 262.
This is gone.
T245. P1 is 1900. Okay, then we were going to try some pistons. Shall we mix them up with PVC and titanium?
Yep.
Try number one again.
Might as well put it, yeah.
One and then 5 or something.
Yeah, because they weren't, when we brought them
Yeah.
up, they were.
Yeah, let's do that. We collect all the others. The other side.
Yeah.
Yeah. Or do you think that they were having this just no?
Okay, we're on piston 1.
Okay.
These can take a bit more, so I'll call this one for at least 500. And starting now. 1, 2, 3.
Can you see it? Can you see it?
J-Rod can see it better than I can.
Okay.
Yeah, yeah.
It's just, yeah, it might have a box inside. So I don't think it's a sink.
Okay.
Site is usually kind of this. That's between 2 and 10. This one's
So, gas tight also?
Yep, take it.
3 of them?
Yeah, like
Okay.
2 on your sampler and a handheld one.
Okay.
Now, they've read one
Yeah,
more piece.
yeah.
First.
Yeah, we'll do another one. Yeah. 33. $37.52 and 110.
Yeah.
Holding steady at 262 degrees with okay, done with that one. With 236. Okay, PVC one would be 5 or 6. 5, 6, and 7, right?
Yeah.
So let's try number 5. Okay, that's it for 5. All right. So 500 on that one again.
Yeah.
So
Okay.
starting piston 5. Temperature drops slightly.
Yeah, so that's so it's the SOI. Yeah, they were, yeah.
Can you see any shimmering?
Yep.
Oh, yeah.
Yeah, it's coming out. Not real fast, but yeah, it's coming out. Okay. See the scale worms moving around in there pretty quickly. I haven't seen many flatworms like that before. Is that a? Do you usually see them on vents around here?
They are not very abundant, but they
Yeah.
have effects, yeah.
Usually there.
Yeah.
This is our combined chemistry sampling and biological observation time. Works out pretty well because we have to sit here for long periods. Okay, that one's done. Tomorrow you can do your gas tights now if you want.
What is the current temperature?
It is 262
Sounds great.
and it's about 50 degrees in the back T2.
Okay, that's good. And the flash pump is running.
Yep.
That's good. Okay. Then I would like to set up for the first gas site. We are going to take one with that is connected to this fluid intake, and it's going to be the one
Gas type 3.
position 3 here. Exactly. I think it should be the valve to starboard. And if you could check first that the right gain is in the system for this one. And for logging, it's going to be gas type 9.
9,
No.
starboard.
You don't? Yeah, starboard 2. That's correct. Yeah.
Looks good to me.
No, numbers look good. Okay. And which sample number is that? The running sample?
10.
10. Thanks. Okay, the pump is still running. Yes, the temperature. Yeah, then I'm ready.
Alright,
Yep.
going to the fire.
Yep.
Fire, what else?
Okay, you can
Yep.
retract it. Okay, good, thanks. Then I'm taking a second one. So
That-
let's check if. Oh, actually, Dave, could you turn on the flash pump again? We turned it off.
Pump is on.
So it's once the pump is back on and running for a little bit, the next one is gonna be
Gas type 4.
yes, correct. The valve 5 pod. If we could also check the gain here before we. And that's.
It's a little low, but
Yeah, do you want to put it higher, or you think that's
I think it'll be fine. I mean,
gas type 6?
I can bring it up, but we'll see how quick it goes. It's not
Then
actually-
the position is 5 pod.
Okay.
Valve 5.
You ready for me to fire?
Just a second, I want to check all the pump is running. Temperature still good?
Yes.
Yeah. Then it can be fired, yes.
I'm gonna have to turn that one up.
Yeah. That's this one. This one we fired before, and now it's this one.
Barely.
Yeah, there it goes.
That stopped the pump.
Okay, that's good. And you've retract. Yep. Okay, that were the two I wanted to fire here, and then the next thing would be a handheld in the same chimney, but I'll let Dave finish first with this one.
Okay.
Thank you.
244.
Is it at all possible, we're getting requests to have the lasers on when we're focusing around, or is it too impossible when we're zoomed out?
That's the most I can zoom in right there within view.
Okay.
You're gonna go back and look at the.
Yeah, the flatworm.
Yeah, that would be great.
We can keep control.
There he is.
Yeah.
Yeah.
Do you think that we would be able to do such a sample here? When you are
Yeah.
finished with
Yes.
the chemistry and things?
Yes, we can do that for sure.
Because I think that it's a great spot there. Like we're seeing a lot of polygon, there are the flatworm, the gastropods, so that would be pretty much all we.
Okay.
Yeah. But again, like when you are finished with the chemistry.
I think we're almost done here.
Okay.
Okay, so we got 5 HFS chemistry samples and 2 gas tights.
I'm going for one gas tank.
I guess,
Yeah, handheld gas tank.
I mean, I could take one more. We're gonna fill everything up.
P2 then, or P3, and we need P2 and P4 as well.
Sometime in the water column, no one knows exactly. And then go down and they go here.
Why do you want? Which one are you picking?
Well, I'm just like, if 1 and 2 are never working fine. Maybe say P1 and P3, and on the other side P2 and P4. You know what I mean? On the
Okay.
it's completely random.
All right. All right. We'll try P3.
You're home. Okay. I'm ready when you are.
Okay, we've got steady temperatures, so here we go. Sampling. Piston 3. Overall sample number 12.
It's P3.
Oh. Hey, so you potentially get 2. That's a good.
Can we check the lights again and see if we can see some shimmering on them? I don't see any.
Very little. Yep.
Maybe a
Some
little.
is very little.
Yeah.
Yeah, there's some. You see, just right at the muzzle.
Mm-hmm. Okay. I'm gonna try something else here. I'll try to add a little bit more. Okay, I don't really see any shimmering coming out on this one.
You don't see?
No.
Okay.
So suspect. But we took enough samples. Okay, so we're done with the fluid sampler. We can take that out and stow it. See what it looks like on the end. Looks like it's turning orange everywhere.
What's your spec come out? Torch is coming out. Right, so well.
Very rusty looking now. Still kind of hot from sitting in the vent.
Are we doing suction first or gas tank first?
I guess we'll finish the gas tight while we're sitting right here. Okay,
You can pick up
stowed away.
whichever seems better.
You're on the flush pump to flush all that hot water out of there.
I'll send out in just a second. One closer to me. You can do that, yeah. Pull it up on the one next to it. It's gonna be hard to see this one.
Okay, I'll go through and label all these guys.
Get off of that angle, and then pretty much you can come towards us if you want, or all the way off to the right. Yeah. You're gonna eventually need to get it that hose and everything off of it. So if you want, just lay it over. On the left. Yeah, on the left. Yeah. Lay it over on the left. Yeah.
Just like here.
Yeah, take a look at it.
If you have a hard time getting it, one leather monkey is orange.
Sorry for that. Have to bring the
This is
forge
gonna
in.
be class like 18. It's just from GGB sample.
Yeah.
Yeah, correct. 1/8, yes.
You could though, use your arm and give me a side view.
Oh, sorry about that.
What's that? 95.
Yeah, GT9 and 6.
Yeah, I need
A little bit of zoom in. And pan left. Pan left in.
The
That's good.
GT numbers were, first one was what?
9.
You guys have
9, then 6, and now this one's 18.
Now 18.
All right, zoom out there.
Our T-max was 294 on IT Pro. Did you get that? You were there.
Well, I was watching the conference.
Yeah, okay. Okay, this is a titanium gas tight bottle we're going to fire in here. This design was by John Lupton, and Conrad Young is the engineer who did most of the building.
Pretty much. It's kind of tilted down there.
With this one, we have to get that titanium intake tube right into the water, hold it nice and steady,
Mayor,
and
would you
then
rather
push a trigger.
me have it pointing out like that and going in, so you can see it going a little bit better, or try to get down up in it as much as possible?
I think down.
Except you don't want to put it into the minerals, right? She doesn't want to suck up any particles in there.
Try to get it as vertical as
Or
possible.
did you get a feel how deep it was?
It wasn't super deep.
Okay, so.
Doug, give me a little bit more distance.
I think that's gonna be good.
You like that? You like that
The
angle going in?
angle is gonna be not just maybe a little back.
Help them come down first. Start going back.
Yeah.
Sure. Alright. You should kind of feel like I was hitting. I feel like I'm hitting right there. Come back a little bit more. Touch more, is it?
Somebody's commenting on how long we've been out here. Yeah, we have been out here a long time. We left port on March 3rd, 26 days ago, and we've still got about 7 days of work on out here before we head back, and it takes us 5 full days of traveling on the ship to get back to Puerto Rico. Okay, we're putting that titanium intake nozzle right down in the vent. Sort of buried in the fluid there. This type of sampler works really well when you can see the vent well and it's a nice clear big opening. It can work well in this particular situation, but you really gotta get it in just the right spot. The tube goes into an evacuated titanium cylinder, holds about 150 milliliters of water, and it's pumped down to a high vacuum, and as soon as we hit the trigger and open that up, the water just floods in from, from nearly 400 atmospheres pressure to a vacuum,
Right there, I feel like I'm hitting.
slams into that bottle, and then we close the valve and it traps everything inside, keeps the gases in there. Get that back on the ship and connect it to a vacuum line. Make sure everything's tight, and then open it up and pull the water out, extract the gas, package the gas up into glass ampules for different types of analysis back on shore. These are going to be analyzed for a bunch of different gases, hydrogen, methane, CO2, hydrocarbons, helium, and helium isotopes. That's the specialty of the helium isotope lab that Tamara runs at PMEL. That lab was established by John Lupton, who was previously at UC Santa Barbara, and then joined NOAA in about 1992, 91 or 92. John retired not too long ago and is still living around Newport, Oregon.
See if we get a tank and we can see it.
Anyway, that helium isotope mass spectrometer is one of just a few instruments like that around the country and around the world.
Let's go and lay it down, but this way I'm directly going up to that.
Would it be best to use the other arm and check with the camera?
The helium is coming out of the mantle. There's
Yeah, that's a great idea.
two types of helium. There's primordial helium-3 that's been trapped inside the Earth since the Earth formed. And then there's helium-4 that gets formed by radioactive decay. Helium-4 is quite common. Helium-3 is not. Helium-3 is only coming out of the deep parts of the Earth. So what you're looking at here, the water circulates through, well, we think it's actually circulating through mantle rocks. And it's also
Then the top left of that screen,
in
you
contact
see the
with
gas
volcanic rocks that are produced by melting the mantle. And when you melt the mantle, components like gases tend to partition into the melt and are enriched relative to their overall concentration in the Earth and concentration in the mantle rocks.
All right, I'm going to zoom out a touch so we can see the radio.
The helium-3 to 4 ratio can tell you something about where that helium comes from.
How's that look? Are you okay with that, Schmidt? Go down the ramp. Go down more.
Yes, please.
We usually look at the ratio and compare it to the ratio in air. So take the 3 to 4 ratio of a sample and normalize it to the 3-4 ratio in air. It gives you an easy way of comparing things instead of thinking about how many moles of helium-3 per, or atoms of helium-3 per atom of helium-4 exist. You just compare it to the ratio in air. So the there goes the trigger.
Is that the max?
Yeah, that's that.
Okay, then it's, yeah. It's probably just the angle that it might look so short. Okay.
So typical mantle helium in hydrothermal vents has a ratio compared to air of about 7 or 8.
Okay.
If you happen to tap into
I'll bring it up to the
a hot spot, a plume, where you're bringing up material from deeper down in the earth, deeper into the mantle, that ratio tends to go up.
What do you think?
Yeah, you know.
Should have gone down further?
I think, I think so that it should have, yeah. But the whole stem
I'm gonna
looks
do
short.
I'm gonna do a test real quick.
Yeah.
Yeah, I pushed down as hard as I could.
Yeah,
It would have gone further. Unless it needs more pressure.
So you're not sure if that one fired?
Yeah.
You think it probably did not, or what?
You know the distance of how far the O-ring moved?
Mhm.
Looks very short. Yeah, I think it got stuck somehow. Yeah, I mean the whole thing looks too short, yeah.
Do you need to adjust the pressure settings again, or
I think we had it.
your gain on that?
It might not close properly. You know that it doesn't retract all the way. Maybe that's the issue because the whole thing looks kind of short.
No, I mean, let me retract that.
I mean that our bottle. Oh no, oh, yeah, that's right. We can check that.
Yeah, like,
yeah.
I mean,
Okay, the distance looks correct.
Yeah, that looks fine.
Okay.
What do you want to do? Do you want to take another one there?
Actually, maybe that's probably the way to go.
Okay.
Yeah. We have two, so we're gonna the next position would be the fence we took samples from yesterday, right? That's the second can set up for fluids.
Yeah.
And we have two good ones from yesterday, so I think then I'm just filling up
All
the
right,
third one.
we've got to get that,
Is that okay, Dave, for you?
Sorry, is what okay?
look,
Tamara
it's just
wants
how
to
to first.
fill up the second gas tank.
Yeah, I
If
just
you
gonna-
want to,
I just
yeah.
take the second one as well, because if we go, our second fluid sampling site is the one from yesterday.
Yep.
So we have two good gas types from there, then we
Okay.
just fill out our third one, once
Okay.
we're there, and we two handheld here. I'm not sure if this one fires properly. but-
Okay. So you'll end up with 4 from
Four
here.
from here and
Okay.
one from the other one, plus the two from last time.
We'll be covering it well.
I got you with the camera.
What would be the gas tank number?
I'm sorry.
The next one,
The one next one. Yeah.
the next one is 11.
11.
Yeah.
Okay.
I don't want to rotate you guys a little bit. Put that other one back on. Sorry. Just rotate your other one. Either way, yeah. Yeah, if you grab that again, pause, yeah. All right, you just want to, like, you're on left. Just y'all left. Show me left. Just y'all left.
Oh, I'm sorry.
Yeah.
I'm sorry.
I think I can sneak in behind you.
Moving left. Freeze this.
Okay, we're moving around to get the gas tight bottle put back onto its holder position and get it safely strapped down again.
All right, Doug, you can put it back in there. I'm gonna, you freeze real quick. I'm gonna come down just a touch more, and then you are good to strap on. You can start working your way down, just gonna get it.
The local time here is just after midnight.
Good. Please. Your way. Clear.
Nice.
Okay, that one is safely back in place.
Get the monkey switch to the other one. I'm gonna slowly start working in the.
We don't want to drop these. They're quite expensive to make, and also actually getting the machine shop that's capable of making them is not an easy thing. Some discussion on the chat about
What's that?
how the animals get from one place to another. Many of them put larvae up into the water column and away it goes with the currents, and with any luck lands on another vet site or a spot that's a suitable habitat.
Yeah, or going right there, or if you grab the rope, you're good. Not
The snail here seems to have a somewhat different strategy of laying its eggs in packets and probably having them hatch right here in place. to know where larvae go, you need to have a pretty good model of what the currents are doing. Sometimes currents are sort of directed along the ridge axis, which will take larvae from one site to another, if those hydrothermal sites are right along the ridge axis or in the axial valley. With seamounts, it's a little different story, since seamounts can be separated and isolated. We found 3 new vent fields here on this trip. One was a volcano that was known, but was not verified to have high temperature venting or any kind of active venting. And that was the first place we went to over 3 weeks ago, or about 3 weeks ago. And we did find high temperature vents there. We're going to get back to that site before we finish, because we weren't able to actually sample the high temperature fluids there. And we only saw one of the chimneys, and we're pretty sure there's quite a few more that are active. So we'll be finishing up down at that site at 20 degrees north. And we also found a vent field at the E-mark area, which is very, very close to another well-known vent field called Snake Pit. But this site that we found was a little bit away from the neovolcanic zone or the ridge axis. And we found it while we were looking for sites of low temperature venting due to serpentinization. Was the main thing we were trying to do out here, is find another site where there's low temperature serpentinization taking place and venting at the seafloor, creating alkaline or high pH fluids that precipitate calcium carbonate minerals instead of these metal sulfides that we're seeing here. We did not find those. We found plenty of other things. Learned a lot about the geology of oceanic core complexes. We did find, as I said, 3 new high temperature vent sites. So the chemistry of those sites will tell us something about what's happening below the surface, what types of heat sources there are, what kind of rocks they're reacting with, what kind of chemical energy sources those different sites produce for the chemosynthetic fauna that live on them. And then the biological observations will catalog what species are living at the different sites and how they're related to species at other sites up and down the Mid-Atlantic Ridge. And when you put all that information together, it can help to create chemical and biological models of how vent systems work. Give us more clues as to how species migrate from one site to another, and also inform us about what effects deep sea mining might have if we take out one of those vent sites, take out the communities of one of the vent sites. We don't think that's going to happen because there are supposed to be environmental regulations that protect these communities along the mid-ocean ridges. But the International Seabed Authority is still trying to figure out how to make those rules and whether to allow mining to happen. It's certainly not an easy thing to come all the way out to the middle of the Atlantic and mine something like this. Most of us who study these systems don't want to see that happen. And I think the general concept for mining on the mid-ocean ridge sites is that only inactive sites would be allowed to be exploited for minerals. And again, I'm not sure that's going to happen either, but that is being investigated as a possibility for getting critical minerals.
Blow down, blow.
So we're gathering scientific data about what's at these sites, how they're connected, how the ecosystems function, and how they are maintained over time. But we're also finding new mineral deposits, so it's hard to know how the information will be used. I think what we found at the Puy de Fol site down at 20 North has changed that site description from inactive to active, and we now know that there are vent fauna communities there and high temperature venting activity. So I think that probably changes at least changes the prospects for anything being mined at that site. Anyway, that's a big question that's being debated right now. The main interest for mining is not really these metal sulfides. They are of interest, but the much easier target to exploit is the manganese nodules, which form right on the surface of the sediment in the deep abyssal plains, where metals are dissolving in the sediments and migrating up to the seafloor and slowly forming these manganese oxide rocks that sit on top of the sediments. There are just huge areas where those exist, like areas the size of all of North America, where manganese oxides are just sitting on top of the seafloor.
Good to start?
Mining companies, I'm sure, see those as a very easy target. You just build a machine that would go down there and scoop them up.
It's not very hot.
In the process,
I mean, it's not shimmering.
raking up the seafloor and
Volcanoes are shimmering, right?
putting lots of sediment up into the water column. The nodules have to be scooped up, brought to the surface, processed, and then the waste material would be pumped back down into the ocean, potentially creating gigantic plumes of particulate matter that would change the character of the deep ocean.
That's good camera view, Doug. Go ahead and start getting ready to action go. Oh, wait, go underneath that. Punch.
Okay, putting that one away. Putting that one away.
Yep.
What did we get up to there? Sequence-wise, 14?
That was
Good.
sample 14, gas type number 11.
I see that. All right, you're going to strap it over. You're going way too far. Shoulder and left.
You gonna go around to the other side? Or back up to the top?
It's time for some biological sampling. Suction sampling. Got some eager biologists here waiting to get some samples.
Patiently,
I'm
give it
gonna.
time to.
We've been ventilating.
They've been sitting here looking at all these little critters and deciding which ones they need to sample. There are some animals here that they haven't seen very often. The black or dark colored gastropods that are living on the sides of the chimneys. We probably already have some of those on that big chimney sample that we put in the bio box. But they're going to use the suction sampler and try to get some of those. There was a flatworm that they're going to try to sample if it's still crawling around down there somewhere. Some of the long scale worms with the many feet. Anyway, I'm going to get out of this chair for a bit, and we're going to listen to one of the biologists direct some sampling. We've taken already 14 samples overall, including 4 rocks and 10 fluid samples. Oh, question about Niskins though. Diffuse flow, takeoff. Okay, those can wait. Yep, one each here.
Almost there now.
You were making it up to you guys.
A little bit?
I can do it.
Trade off.
You all right?
You can both talk in the microphone too, so
Now pitching.
we can easily hear you.
Right now?
Happy symptom.
Alright, so we're starting with the Platyhelminthes.
Right.
Hello.
Almost.
Yeah.
Yourself have a view of it.
Hello, I'm Joan Alfaro, postdoctoral fellow in the University of Victoria. In Amanda Bates' lab, I work with ecology of hydrothermal vents and functional diversity, and we are now going to try to sample some animals that we are interested in with the suction sampler. Think that we will start with flatworms that we have been observing. We are interested in these animals because we want to test several ideas we have. We have been observing egg capsules here and there in this hydrothermal vent, and we are not sure if they belong to the gastropods or to the flatworms, and so that's why we are interested in sampling that. Number 1, it's fine. All the others are like big mesh, like number 8. So we can use
So only use number 8.
2, 3.
Yeah.
So start with number 2.
Yeah.
It's fine, yeah, in the beginning.
And we won't, it's all the same sites, we don't mind if it's like, just keep adding.
Yeah. If one is working, we can put everything together. Other animals that we are interested are the black gastropods that we see around these chimneys. Those guys are very interesting for us because these kind of assemblages have been only recently described in the Mid-Atlantic Ridge, and so we can still obtain a lot of information. All samples are gonna be divided between a bunch of biologists for studying a variety of things, including viruses, genetics, population genetics, taxonomy, etc. So many exciting questions that we can answer by sampling all these animals. Then we have been observing also some very interesting polychaetes, some very interesting worms that are kind of reddish and long, and they move relatively quick. We do not have any idea of what it could be. We suspect they could be a family of polychaetes that are relatively abundant in some places, but we really need to check it under a stereo microscope to make sure. Okay, it seems we are ready. Let's see if we can locate the flatworm we were observing some minutes ago.
Ready?
No, not yet.
You're in your element.
Hi, Jason. I'm reading now all your comments in YouTube. So if you want to ask questions about biology, but yeah, be good. I'm an ecologist. So yeah, I do not know everything about these creatures. Usually ecologists work more at the community level, so I'm more interested in the interactions of these species between them and between the environment. But yeah, go ahead. So other things that we have been observing is another species of gastropods that hopefully we will be able to see in a few minutes. Those are also black. They are black basically because they are covered by minerals that precipitate from the fluids. In reality, they are more like brownish. And those are very small gastropods that kind of hide if you do not have a trained eye. So at least we have like 5 or 6 animals that we are interested in trying to sample. From the point of view of ecology, what it's really intriguing about this site that we discovered only a couple of days ago is the high abundance of gastropods, especially the big ones that you can observe that are more brownish. Those are usually scavengers and predators. They belong to family Buccinidae, which are relatively common in peripheries of hydrothermal vents, but what is really amazing is the abundance that they are showing here. So that's something that we are very interested in, and that's why yesterday, 2 days ago, if you were following the dive, you will know that we collect a lot of those guys, and it's pretty amazing how they could be so abundant. If you remember the vents that we discovered at Kane Fracture Zone, these gastropods were much less abundant, and what it was really abundant there were the anemones, the white anemones. There are some of these anemones here, but not close to the abundance that we observed at Kane hydrothermal vents, and it's this kind. Oh, it's okay. It's these kind of questions that we ecologists try to solve: why species occur where they occur, and why they present the abundance they present. And this is kind of like, yeah, the broader questions we really are interested in.
Was it the dead? Was it the dead shrimp?
And I think the first thing you said was 3750.
That was the dead shrimp.
Right, right. Okay. So we're pretty deep on it. Go the other way.
This is a bio.
The one yesterday was 3734. So maybe when you're ready to go back that same direction you swim. So, yeah.
Okay, we are now waiting for the slurp gun. We are experiencing some technical difficulties. Hopefully we will fix that. If not, we have other tools that we can use. But as you all can imagine, sampling in the deep sea is very challenging from the technological point of view. These ROVs are really masterpiece of technology, but they are constantly being tested and challenged by us, by scientists, by asking all the time for samples here and there. And so yes, I'm reading now comments. Yes, Jason, they're probably limpets. We think we observed some limpets at Kane hydrothermal vents, but we didn't have time to sample because they were relatively sparse, so not very abundant, not as the gastropods that we are observing. So yeah, there are definitely a lot of limpets at hydrothermal vents, not as abundant maybe in the Atlantic as they are in the Pacific, but still, especially when there are mussels, they kind of like a lot to hang over the mussels and graze the shells of mussels to eat bacteria and these things. But here they are kind of less abundant. The polychaetes, the tube worms that we are observing, those are a species that is very common around hydrothermal vents in the Atlantic. They are basically filter feedings. They only show two pulps out of the tube, which they use to take food for them, and they rarely go out of the tube if yeah, okay. So they almost never get out of the tube, and they like a lot those places where we find hydrothermal sediments, or sediments that has this kind of reddish color, and they come into the vents only when the activity decreases a bit. So here, if you check all the animals, you will see that they are not that abundant, but as soon as we get out of the vent, they start to be everywhere, basically. Yeah, I don't know how that shrimp entered, but I'm not sure they are inside the jars. Maybe they are only hanging around. Yes, we were able to ID the mussel shells that we found, and they definitely belong to Bathymodiolus mussels. Bathymodiolus mussels are a genus of mussels that inhabit hydrothermal vents in the Atlantic and are those kind of animals that present a symbiosis with a bacteria, and so they kind of do the same thing as the shrimps here. They locate themselves in areas where they benefit from hydrothermal fluid, and they just filter this fluid, send it to the branchie, to the gills, and in the gills they present symbionts that will do chemosynthesis and then pass this energy to the mussels, and so that's the way they grow. So at some point, these hydrothermal vents had some mussel populations, but for whatever the reason that we don't know, they are not here anymore. So a lot of questions that kind of what we are producing in huge quantities in this cruise, a lot of questions, which means that it has been a very good cruise so far. So very happy. Those mussels or these genus of mussels also occur at ships, but not necessarily the same species. So yeah, it's exactly the same genus of mussels.
We're going for a flatworm.
Yeah.
If it hasn't run away, it was moving really fast.
Yeah. So we will try now to go for the flatworm because there are not a lot. So if we can make sure that we. Yes, here it is. That guy.
That might be the other one. That's two of them. There were two. Just gotta find the second one. The second one's hanging out over here, right?
The second flatworm, I think it was next to the metal piece of the ROV.
Well, I'm talking about the first one.
Oh, the first one is, I think it's down there. It's the one that you focus.
Oh, you think so?
I think that it was that one. I think.
I just didn't see the other one.
Yeah, I've only seen two, so probably it's that one.
The other one was right here.
Yeah, yeah. The other one was sitting around here, and I don't see it anymore. Yeah.
Sean, should we try with the Alfiora as well?
Yeah.
You're gonna porch out as you go over there. Just go a little bit more around. And the zoom in and suction is coming on now. Might be able to bring my rocket in a little bit if you could. Let me get you a little closer to it.
Okay. We have so much rims in, unintended sampling, but okay. It's always good to have some extra material. It seems that it's very attached to this rock. Let's see if we can get it.
Come out a little bit. Yeah, bring your arm out a little bit because I'm going to bring the ROV a little bit closer.
We are now gonna position the ROV slightly different, so we might be able to have a better angle, and using the slurp gun again, we might succeed in retrieving this flatworm. The purple sock monster worm. Whoa, I'm not sure I know what was that. Yes, come on, keep there. Yes.
I haven't seen the shrimp come through yet. Oh yeah, they're there.
It's in this. Yeah, here it is. Good one. I saw it like poof. Yeah.
Section's off right now, so.
Perfect. Amazing skills of our pilots here.
More in there, or do you want to index?
That's sample number 15.
Yeah, you can put more in there.
More in there with
Yeah.
the worm?
Yeah. Yeah.
Okay, so,
I mean, is it?
what's that? Imagine they want another flatworm if it's round, but I'll guess not.
Yeah. If not, then we can
The ophiuroid, is that what you're adding next?
we can add the ophiuroid. Yeah, it's just there. Yeah. So we are looking for another flatworm. We only see 2 of them so far, but probably it's already gone. So we are now gonna try this ophiuroid here, a brittle star.
Just below the stuff you said. Oh no, that's the snail's foot. Never mind.
Oh yeah, yeah. Oh, but I thought it was. Yeah. Okay, let's see if this guy wants to come out.
I think I saw him go in, but
Yeah,
not positive.
it's okay.
I'll keep an eye. Hold on, I'll keep an eye. Alright, rocks just came in.
The shrimp is over there.
Nope, I don't see it.
No, it's okay. It's okay.
So, polychaetes next?
Yeah. So if we can focus in, do you remember that we focused on an area that were some black gastropods and a lot of like reddish polychaetes? I'm not sure where it was. I'm not sure if it was. So we are now gonna try to sample some of the worms we have seen. Those are reddish long worms, kind of a mystery for us. We have some ideas, but not exactly an identification for these guys, and that would be a very valuable sample for us. One of the advantages of doing the chemistry first is that it allows us to focus with this great zoom that we have a lot in all the animals around, and so in the end we can take our suction sampler and start sampling all the things we have observed so far. So you can see it's a very dense community, so it takes a while to discover all the different creatures that are around. They were more with the big black gastropods, and they were really close to the chimney, so I don't know if those areas there
It looks like the shrimp took over that area.
could be. Yeah,
That's where I went first.
yeah, these areas here.
All the worms ran away. All the worms ran away. Probably all.
Those polychaetes are very mobile, so in very few seconds they can move a lot.
The long red ones, right?
Yeah.
Yeah.
Long fast ones.
Shrimps a bit everywhere. Okay. I think it was around there, but yeah, I mean they move a lot, so is this? Oh no, that's the food. They were here, right? Yeah.
Oh, zoom. Yeah, there's one right there in the center of those gastropods.
Oh yes, here it is.
Alright,
So-
center screen. Huh?
So
We
what?
just pan to the left just to see if there's more in this other, like, because if there's more, we might as well go for the one that has a few rather than just the one way together. Yeah, I'm not seeing any other spot now.
Look at the center of that cookie. The little guy,
We-
right there, and this one is going away. See that center screen?
Yeah, perfect. If we, yeah, we can do one section here and take the gastropods and the polychaete.
When you get there, oh, here we are. And then section is on. Right there, and I just want to get this. The little guy, the, right there, and this one is going away. See that center screen? Yeah, perfect. If we, yeah, we can do one section here and take the gastropods and the polychaete.
Yeah,
And then section is on. right underneath that one picture. Yeah.
Good. Good.
You're pushing us pretty hard. Try to use the bottom part of it to like scrape up on it.
Yeah, perfect.
It's going into the same.
Yeah, yes. We got it.
Same sample.
We got it. Perfect. I already said that, but we have very, very, very skillful pilots that are helping us
Up there on
tones.
the top left near the tail of the shrimp.
Center screen dive?
Yeah.
Yeah, we have spot another one, so let's keep sampling here. Those are these small.
There now.
Perfect. Yes.
Yeah, so there's just another one just further up.
Yeah, stay up just a touch. Just left the center screen right behind the stuff. I'm gonna porch out a little bit, give you a little bit more reach. There you go, you should be good to go.
So as you can see, this will be a very diverse sample with a bunch of different animals that hopefully will increase dramatically our knowledge of this site, as we will be able to dissect these guys, observe them under the microscope, and do a bunch of analysis.
Over here, go.
We,
Just left the stick. See it?
yeah, here we are. That is what I was telling. As soon as you start observing all these areas, you start to find more and more and more animals.
There's still one.
Come on. This one is very stick to the rock. Ah.
Should go in that way right there. Push me off.
Yeah.
All right, they're fine now. I'm stable now. No, I'm not quite there.
Okay, we are stabilizing a bit the ROV, and we're gonna come back. Oh, now I see another one over there. Yeah, hopefully they get along in the sample tank. So far we are not collecting that many species that are potential predators, so probably they will get along in the sample tank. Oh, there is one down there.
Yeah, down below.
Here. Yeah. Okay. No, but it's good that we are already collecting gastropods, so. Mm-hmm.
I'm stable again. No. Right there.
So this guy is trying to go away from us.
What you got? Like that and turn suction off.
Oh, okay, now it's trembling. Yeah. Now it's almost there. Now it's almost there.
I'm gonna turn it off for a second, and then I'm gonna hit him with everything I got. Off. And off.
is there still?
Try to rotate your jaws clockwise a touch. Keep it there, yeah, push in as you rotate clockwise.
Yeah, we can try to collect these snails and maybe it comes with them if we scratch. Yeah.
Just trying to leave it there. We're free without doing too much.
Any more? No scan?
Okay. We might, yeah. We might try to scrap them if we collect all the gastropods. Yeah.
Get those gastropods and
Yeah. We are interested in these gastropods too, so if we collect all together, it's perfect.
turn it up a little bit and just, yeah, like that.
Yeah, perfect. So we're gonna try to collect all together now, scratching a bit the rock. Yeah.
No, sorry. You're all right. All right, we want to go back in that same spot, right?
Mm-hmm. Yeah, we have a lot of hope in this very big chimney we collect because we already spot some gastropods, so who knows, maybe it comes also with some of these polychaetes and maybe other unexpected animals. That's gonna be a challenge for us to check all these pieces of chimney that we collect. We are now again stabilizing the ROV, so we have a good position to try to sample a batch of gastropods and hopefully some polychaetes. But as you can see, it's a very steep terrain, full of black smoke hairs here and there, so it's not an easy job. And despite of that, the pilots are doing an incredible job here.
Tight for you, but like that.
Okay, here we go. Oh, I can see it there. Yeah, let's scratch this and yeah. Now.
Still get a little force on there.
I've broken another rock. Let me get on top of this thing. Just keep shattering these things I'm trying to sit over.
Okay, so as you can see, there is a lot of smoke now. We will try to stabilize a bit and go again for the red polychaete plus some gastropods. These are one of the 3 species that we are seeing in general in this hydrothermal vent. As you can see, they kind of inhabit only very close areas to the fluids, the black smoke that is venting from the chimney, and this kind of tell us that these guys could potentially have a symbiosis with bacteria. This is something that we will definitely study in more detail with the samples that we are collecting. You will see also in few minutes one of the other species, which is much smaller. That one probably is not symbiotic, but we will also check it. And then you have like the very big, large gastropod that is all around this hydrothermal area that we were talking before. That this gastropod is a predator, so that's why it inhabits more the periphery area, although it comes relatively close. So this is basically what we call a zonation, where animals distribute themselves according to their environmental preferences. It is pretty illustrative to observe this bend and explain and use it as an example for zonation. Here we go, the polychaete. We have located again.
On the gas tanks, yeah, just rotate counterclock. There you go.
Oh yeah, they're breathing there.
Okay, 3 polychaete we have here. Let's see. Okay, 1. Perfect.
Oh, sorry.
1 polychaete is in the jar. Yes, the shrimps are covering everything, and it's very difficult to sometimes when they cover the snails to figure out where we observed something that was very interesting and we are now interested to sampling. And it's like, well, probably there are shrimps over there, so that's why it's difficult. Plus, as you can see, they are very mobile, so in just a few seconds they can cover areas that were, oh, come on, almost there. So yeah, Connor, we suspect that a lot of the polychaetes we observed in the previous dive are exactly these ones here that we are sampling. Other ones we know that are different, but those we already have some samples. So yeah, hopefully in the next dives we will be able to give you an update of the identity. It's incredible how small these guys are, but how they really get attached to the rocks in a very strong way.
They have so many legs, they can hold on.
That's true. Okay, almost there. Yes, perfect. Okay. I think that if we finish sampling all this patch of gastropods here,
Now they're coming back out.
we're becoming experts in sampling polychaetes.
Yeah, so if we could just snag the rest of these black gastropods here in this little patch, and then I think we want to get some small gastropods.
The very small ones, yeah. Yeah. Okay, here we are. Those I remember that were very difficult in the last dive as well to to sample. The small ones are much easier to. Perfect. Yes. Here we go.
Quite a bit in there.
Perfect. Perfect.
Give your jaws a shake. You just rotate back and forth, yeah, just like that, fast. There you go. Stop suction for a second. Keep doing it, 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 and max suction.
Good.
See if anything else comes through.
So we have here in the control room many different cameras. Some of them are pointing directly to the jar, so we are able to see what we are sucking is ending properly in the jar, and we are seeing right now a lot of animals coming in. So yeah,
let's switch jars.
yeah, let's switch jars. So we are now gonna change jar because this is already full of different animals.
You want to be on jar 3, right? Go 2 the other way.
Yes. Not jar 1.
So I can tell from here that yes, we have sampled several black gastropods, some shrimps that come with them, and then we have like the polychaetes. And we are now gonna go for the very small species of gastropods. I think
I'm gonna have to move to get where y'all want, right? Let's have a look.
let's have a look and try to locate these very small gastropods. You really have to zoom in, they are very difficult to spot.
See a brittle star, we'll snag that as well.
This will be by far the most diverse biosample we have. But potentially it's gonna be a very interesting one as well, because we have now many questions for many different animals.
We were playing Volt Smash for a little bit. Does that mean that exact area?
No, it does not. It just needs to be the size of it.
With that fish?
Okay, let's see if we can spot them. Here I see a bunch of black snails, but those are the larger species.
So
No.
there's a lot of dead bodies around.
But I remember that they were relatively close to
You want
those.
that one? You want that one? It's a big red one. Almost center screen now. Boom.
Oh,
See that gigantic one right there?
yeah, we can do that.
It's up to you.
Yeah, definitely.
Right down there. Come on, get off.
Thanks to J-Rod, one of our pilots that has a spot here, a very interesting red worm. We will try to sample this.
Let's try to get your nozzle on camera. Just like you are.
Yeah,
I'm trying to get you close.
this is over the mold, so probably it's gonna be easy. Let's fingers crossed.
Why are you gonna say that?
Yeah, why I'm saying this?
That's gonna be way harder. Alright, hold on. Alright, that you could move it and try to do that.
One small stream starts to move, and here we go. I told you, it could be easy. Oh yeah.
Do y'all want more?
Look, there's a big one down there.
And it's SS3.
Do y'all want,
Feed off the dead bodies.
do y'all want more of those things? Because there's a lot.
There's a lot, yeah. I'm seeing it everywhere. If we can collect one more specimen, that would be good.
What are these down here? These everywhere. What the?
Wow, that's a good collection of polygids, yeah. Those are different than the ones that we were collecting some minutes ago in the chimney. You can tell that because the body is not as long and thin, and also I can see several features that really make me think, aside that they are in a very different environment, they are not that close to the fluid, so probably completely different species. What it is, I don't know, I cannot tell from here. This is something that I will be able to explore this morning after using the microscope. So let's see.
Here come the carcasses.
Yeah, we're collecting a bit of carcasses here, but it's okay. Some worms as well.
Oh, look at that one.
Yeah, the one is purple one. Oh,
I want to be bathed.
let's see that. Come on, almost there. Here we go.
Oh, it's beautiful.
Here we go.
What? Yeah. I was from the beginning.
Okay, good. Yes, there are anemones here. Yes. Yeah.
Oh yeah, go right there.
Go slow.
It's gonna try to get these brittle stars as well. Come on. Yes. Some gastropods as well. Almost there. Oh, so guys are strong.
Got one loose. You don't want to get sucked in.
Alright, I'm gonna section off for just a second.
Gosh, some of
Section
these.
off and section.
Juan,
Mhm.
the size of the polychaete.
Yeah.
Right hand side laser just above.
It's big, it's big.
Big.
Those are very big polychaetes. Okay. Should we?
Oh,
Perfect. Perfect. Okay, let's go for the small gastropods that we end here.
beautiful. Yeah. Did we get enough of the
Yeah.
polychaetes we think?
Yeah. I mean, it's never enough, but at some point we have to cut. So
YouTube just popped in. Let's get inside.
This is fun.
Everything.
no.
But I think we have to.
Everything.
Oh my gosh, someone just said breakdancing brittle stars. Oh no.
So now we are looking really for the tiny gastropods that are usually next to the larger black ones. And we seen it for a moment where we were doing the chemical stuff, but I honestly cannot locate it anymore. But it has to be there.
Is Dave going?
Dave's right behind you.
Oh,
Yeah, Dave, I think we're gonna have to come up, so.
Oh, what's going on?
Our submotor is raising, it's way too high. Something's transferring into it.
42 degrees.
No, it's at 97%.
Right here. Right here.
It shouldn't be raising like that.
okay.
Oh, is there anything you want to sample real quick before we come up?
Just if we can locate these very small gastropods around, but if
Sample
it's
real quick before coming
too
out.
difficult.
Quick? Nothing quick. We got a large volume bag and a couple of filters.
Alright, well, let me see if I can find these gastropods real quick and then we'll pop
Yeah.
up.
I think we're closer to the active site.
They were, I mean, we have focused on them at some point when we were doing the chemical stuff, so they have to be there.
Do you want them in a different jar?
Yeah, that would
Go
be.
ahead and move it to the next number. Next, yeah, positive one. There you go, yeah.
Yeah.
4.
Perfect.
It's okay.
Okay. So we are gonna come back here where we did all the chemical sampling and the first biosamples and try to locate these very small gastropods.
And we're gonna be going all the way up anyway, so we might just wait and see what happens. Yeah, it's not like it's leaking.
No, yeah. Strong, that's, I guess, the lower suction is pulling it through the cable.
Alright.
They need to be here. They have to be.
There are
Oh,
some
over
right by
there.
the shrimp.
Yeah, over there.
On the left-hand
On
side
the
of
left.
the shrimp swarm, they're really small little nubs.
So you're getting too hot, you think it's...
I think we might be fine. I have a little theory. We're gonna test it out real quick.
Is it
After we get these samples, we're gonna have a look and...
This is the pump of the.
What's that?
It's the pump of the suction sampler. We see it.
Definitely not the suction sample.
Okay.
That would be amazing. Problem. So let's bring it out right here and do this. Bring it out.
Mm-hmm. It's okay. Huh?
No, it's not heating up. That's not heating up. No, it's a complex filling, which isn't good. Stops. Let me just get the sample real quick. We'll have a look at it.
Mm-hmm.
So she said just left the strips.
I think they have to be.
They're right over here.
Yeah. No, more to the left.
More to the left?
I think.
So.
Okay. Oh, yeah. A bit more to the left. Just
No,
below
to the
this
right.
edge rim.
To the right. To the right, pan right. Yeah. So those little nubs that
Where?
the shrimp are on right now.
This here? Those here?
Yeah, are those the smaller ones?
No. Those are not the small ones.
Oh, yeah, okay.
It's more to the left.
Yeah.
Think. Okay, if you zoom there, just there. Yeah. Straight. Yeah. Those guys.
There we go.
Those guys. These very small ones is the ones that we are interested in.
Well, there's a different species there. When they zoomed in.
I think it's kind of more exposed. So these are the very small gastropods that we are also interested in. In the previous bin at Kane, we found a pretty dense assemblage of those together with the larger ones that we sampled before. So right now, if we sample here, we will have the same animals from different locations, which would allow us to do a bunch of comparisons, which will be very interesting. Perfect, those wise. They're, as you can see, they are very small and difficult to see.
They're covered in that stuff. Maybe rotate a little
Yes,
bit.
that's a section same thing for.
This is bio-cemetery.
Suction strong ones as well. I think you have to bring the arm in and come at a different angle.
Almost.
So,
Yeah, like that. Try to get to the dome on this guy.
Okay, let's see.
It's a hard angle.
Oui.
somewhere, try it, maybe on the eighth.
Yeah, watch your arm with that and smoke them.
Okay, thank you.
You can tell off. That's them up there too with the worm, right?
Yes, exactly. Another patch. I guess that the patch has been covered by a bit of.
Right there, so right where the worm is.
Yeah. If we kind of scratch a bit all of these, I think they are gonna come out.
Look, there's a more coiled kind of brown shell right beside.
Yeah, I think it's all the same species, just that there are individuals that are bigger than other.
You think so?
I think so.
Because they don't have the same coil.
Oh. We're almost there. There are some red polychaetes again in between. Yeah, all of this is like a batch of them.
But like the shrimp on the left, right below it, there's one with a very distinct coil that doesn't look the same.
I think you're gonna have to rotate a lot. Bring your arm out, rotate counterclockwise, almost like you gotta get that hose on the other side. Keep going, rotate 180. Yeah, we'll let you go.
Yeah.
You can try counterclockwise. You're just gonna block the view.
Yeah. Yeah.
Okay, try going in like that.
Okay, here we go. Oh, we have one big one.
Yeah. Push in just a little bit more right when you make that divot.
Yeah,
Start at the divot.
we kind of perfect.
Yeah, go down there. Yeah, go down and slide it across. Down, put some there.
Yeah, they were much easier last time. Actually, the large ones were the difficult ones, and those kind of like just detach very quickly.
It's just the angle that we got.
Yeah, probably.
I think it did, but we can go on that spot. Just gonna look to see if I can find some at a better angle for you.
Yeah. So I feel that if we can scratch from below, they are gonna.
Somewhere in shrimp. Shrimp gonna look right around here.
Are these them?
Shrimp.
No. This is like the batch that I've seen so far.
This whole area right there, and then like way over here.
Yeah, yeah.
Hold on, let me look at this page. It's really crazy.
It's like, one big bio box and one small one. That's one word. There's gotta be like almost 100 pounds of stuff in there.
Which is normal, maxed out.
Oh, okay.
Okay, let's see if those stay there. If everything balances out.
Okay, so here trying to collect the last of the animals that we are interested in, which are these very small gastropods that you can see in front.
Either that, or I can grab the base of your thing and get a better angle. Well, let's try your first idea first.
Over there.
Just get on the base, down a little bit lower. I'm going to start at the top up there and scrape it down to it. Alright.
I was a little surprised.
These ones up here, right?
South.
Okay, we are trying a slightly different strategy now, scratching from above and then sucking it from below with the suction sampler. It's working. Yeah. Okay, perfect. Yeah. Yeah, if we can go, yeah, test those here. Yeah, these small ones, yeah. Okay, beautiful. Yeah, those here, that would be perfect. Okay.
It isn't scraping it off as it's coming out. It's wrapped up there. It just breaks apart.
I think I was just going to see if there was anything right behind your arm.
That's how they describe it.
I don't see anyone right there.
No, all of them are in front.
You want any, is there anything else right here? Am I missing anything?
No, it's okay, I think.
Okay.
Perfect, thank you very much. So that has been a long biosampling,
Thanks, fellas.
full of animals, our jars now, so a lot of material. I leave it to our chief scientist, Dave Butterfield.
Move it one more. Positive number? We'll have a look back. Hold on real quick. No problem.
Now J-Rod's gonna figure out what we're gonna do.
Come back real quick.
Yeah, over here.
Yeah. Sorry, show you. Oh, perfect.
So what's our situation?
Two, go, three positive. It looks like we're leveled out. It looks like we're gonna be all right.
Okay, what was going on?
So, one of our comps was transferring it to the other one, but it's leveled out now. So the other one's not dropping anymore, and of course that one can't build. So we're gonna monitor it a little bit, make sure it doesn't spike any higher than what we're at, but it wouldn't make it any worse going up.
A little more.
Yeah. That's what you got.
God, that's a lot of animals.
I have a very good list here.
One more time.
And then I see all the small ones. Yeah, perfect.
Okay, yeah, we
Awesome,
go
thanks guys.
negative, what, 3?
Look at that Betty. Look at that big.
Yeah, but so go negative.
And those are not scale worms.
Yeah, keep coming.
2.
Yeah.
Those are all the things that I'm not sure.
One more.
Okay.
That's
Okay.
much more than what I was able to collect.
Go. That's a great question.
That's
Will
great.
you log in?
Can you tell me?
I usually.
Perfect.
Okay, well, if we're able to stay, next thing is diffuse fluid.
Term is,
Some of it's coming down now.
Wow, that was incredible.
no, I'm using it as much.
See it? It's dropping.
That's like,
Yeah, because I'm not using it as hard.
that's
So
the
it's gonna
list.
peg out when it gets hot.
That's
It's
the
52.
wish list completed.
It's dropping.
Because I'm cooling off down there. I was using the septic tank right now. So as I do this, it's gonna cool. So you'll get just hold on to that for a second. So let's just have a look.
These guys are there.
Oh, they got that.
Okay. Stop again.
Alright.
It's like there. Good. You got your. No, there. Let's get it. For the memories. He's a champion. He's already famous. He got Twitter famous. And now he's on
Yeah, so that allows it to drop a
I
little
would,
bit
I
more.
would have been, and all of a sudden, I start receiving that notification. And probably that's why it's not receiving.
Term is staying inside.
Yeah.
I think we're good. Let's keep an eye on it. All right, Dave, what would you like me to do, sir?
And all of them, for sure.
Yeah, yeah.
No, get rid of this.
Do you need a plug?
Yeah. We're trying to go inside that. Close. That's what we're doing.
Okay, do you need a camera? Okay, fine.
That's it.
All right, where would you like to go, sir?
We are looking for some diffuse flow in a shrimp pile to do microbiology sampling.
And these shrimp right here?
Well, maybe. They don't look very active. Although I
Want to do the
guess
active ones? Like these ones over here?
But I wanted the big ones for the power.
Can I bring the porch in?
they're looking for something, you know, maybe 15 to 25 degrees range.
In a second, we just finish stowing everything.
Mhm.
Like everything. All right, cool. Stowed away. All right, so looking for some diffuse flow, huh?
How many biosamples was that, and what number are we on now?
So there's 3 logged as 3 bios,
Mm-hmm.
15, 16, 17.
Okay,
The next sample is 18.
now on 18. Okay. All right, now we're looking for a spot with a bunch of swarming shrimp.
So good to move around up from this, right?
We can look around, yeah. But they wanted something in this, you know, down here on this side of the chimney, as opposed to all the way up to the top of it.
There's one right up there, see it?
Yeah, yeah, that might be good.
Or
That's the one? That's the winner? What do you think about this guy?
Yeah, that could. What do you think? You like this?
That looks pretty good.
Look pretty good. All right.
Pretty good,
Try
or-
try temperature measurement away from the black smoke. We don't want to be in the black smoke, right?
Yeah.
I mean, if
But
I come
it's down
up,
below.
it looks like there's like a million shrimp on that other side.
Mm-hmm.
Like it's like shrimp plates. Like
Yeah.
that's just nothing but shrimp all the way over there.
Shrimp City. Yeah,
Shrimp
Shrimp City.
apocalypse.
So do you have a specific area you want to sample some shrimp water?
So we just need some shimmery flow, basically, you can see around the shrimp. So we could go down to somewhere where we see some shimmery flow.
Roger that. Let's for shimmery.
Shimmering shrimp. We're looking for shimmering shrimp.
Shrimp style. Wood style.
Can you pay me in about 10 meters? I'm not seeing any shimmer in that.
Depth here, 30. Depth here is 3747 meters.
That doesn't look like shimmer to me.
That looks kind of like flax.
Am I good to go out and around to that other area?
It's up to Mark and Sabrina. How far?
Are you good
How
if I
far
go
we
out and around this black smoke right here to the other side?
Yeah, that sounds good.
Roger that.
Oh, lots of smoke.
Peeks my left real quick. Make sure there's nothing tall over here, there's nothing running. There sure is. Take that out.
We have not fired any Niskins yet. We're about to try to do diffuse sampling. And, you wanted some by the diffuse and then some in the smoke plume? Okay.
Very nice biology samples.
Yeah, they got great biology samples. So we're gonna fire 1, 2, 4, and 6?
Yeah.
Okay. For a while, it looked like we might have a problem and have to come up, but we
We're looking good right
seem
now.
to be okay. All right.
It's still not, it's not great, but we're.
Okay. Well, that's a lot of shrimp there.
I'm not seeing any diffuse though. Are you seeing, let me know if you see anything.
I think it's pretty neat
Like
that
here,
we
maybe?
can say,
Oh no, maybe that's
oh yeah, we're just starting to do the sequencing. We'll figure out what we're We'll figure it out. By we, it's Sean. Sean Takami's there. Just so. It's a DNA detector.
Should you just give it? Yeah, they want the clean shimmer.
Oh, there's some.
Done that.
Right here?
Alright, I'm gonna come around.
Oh, there's our marker. We're back up where we were.
There's definitely some shimmer on this side. Yeah. So do you want to be on the same side of that, or do you want to be somewhere new?
Is this the same side?
We, the marker is where we took the samples the last time.
Oh god.
Yeah.
So
So
we've
I
come
can
all the
go
way
find,
up there.
I can come find you another one though. That's not a problem.
That's so funny. Ideally, yes, but if we don't have much time, then we could sample there again.
No, I'll find you something better on the other side.
J-Rod aims to please.
fine, just fine.
Where are the sharks at?
Right.
Has to be some shimmer here.
I mean, if you see shrimp that are swarming around, there's probably going to be warm water in there.
Yeah.
It looks so, yeah, it looks crazy. Yeah, smoky in the other cameras. Must be some shimmer over there somewhere.
There has to be some.
Right up here to my left.
We could just put a temperature probe in there and see what we got.
Yeah.
Yeah.
Just have a look at this little area right here where the rock is bare for some reason. That shimmer right there?
Looks like.
Yeah, it looks like a little bit right at the top of the screen.
Yep. And also here, both places here and here. Sample of the rock.
Okay.
Yeah, both of them.
Alright, so good to land out in this area?
Hmm.
There's such a good graveyard down there.
Yeah.
Put me in another
All
tank.
these dead shrimp.
Roger that. Vehicle's stable out right here. Just verify we got sugar.
Okay, so we are trying to get a sample of diffuse fluid in an area where shrimp are living and looking for temperatures around 15 to 25 degrees. Trying to get a large volume of that so that the microbiology team can do it's called a stable isotope probing experiment, which is essentially incorporating heavy carbon isotope, just C13 carbon isotope, into DNA and RNA during a culture experiment.
You don't want to do temperature probe first, right?
We could do the temperature probe first, or yeah.
That way we know it's a good area.
Yeah, then we could make sure that it's not too hot.
Be all right. Maybe a little tangled up, so you have to drop it down a little bit. Yeah, there you go. So we bring the torch in.
Track.
Where would you like us to probe it?
Just you can just go in anywhere where you see shrimp swarming.
There's some down
Down here, center screen, Doug, that hole. No, center screen. Sorry, we put a laser on it. Left laser.
Okay, checking the temperature here.
Went up a bit. We're at 4 degrees.
Mm-hmm.
That's about it.
Yeah, not much there.
Need more
I
fun.
would try putting it in where the shrimp are most active and just see what you get.
Yeah, I can go over there. See if you got anything within your reach there. Show the right, let's see if there's anything which side with a sport of right back on that corner.
Dave,
Yep.
what was that temperature there?
Oh, just 4 degrees.
Just where they're moving a lot, instead where they're swarming a lot.
That's really
You're just like perched up and just lean up against the water.
taking shrimp water.
Yeah, trying to find some
That's 10 degrees, 11.
might be a good spot.
Well, there's a couple of shrimp. One, maybe two.
Just a couple.
Just the right.
16.
gosh.
Okay.
It's hot.
Yeah. Where is that? I can't. Okay, it's in there. Okay. So we're getting almost 20 degrees right there. It's with almost 20. It's reading 13 at the moment, but we got 15 to 20.
Morning, sir.
Good morning.
How are you today?
Oh, pretty good.
I see we're on the back side of this thing.
Yeah,
it's
yeah.
a
We
lot, huh?
started down at the bottom.
There's a few
Came
shrimp
up.
on this thing.
Yeah, I'm not a biologist, but I'd say at least 10.
More, a few more than 10.
Maybe we could do like the jelly bean thing where you guess how many you win a prize or something.
Mm-hmm.
To have some sort of AI count them all. Because it's like, there has to be thousands and thousands.
I already counted them, I just don't want to tell you.
Oh.
You'll have to guess.
So it isn't just 10, is it?
No.
I wonder what the weight of all of the shrimp, the biomass.
Mm. Not super warm there, but what about right below? In this camera, there seems to be a lot of movement right below basket.
Yeah, I saw that.
20, close to 20 degrees. Okay. 20 and a half. Okay. That's not bad. Oh, down low in front of the basket, below that bare rock there, and
This kind of neighborhood right here?
yeah, somewhere in there. Mm-hmm. Tried a hole, and it did not have much temperature. That's 4.
What kind of temper you hoping?
I mean, 20 degrees would be fine. So we had that at the other spot. Just wanted to check what else we have.
I wonder about that rock. Because a lot of times it's affecting that much.
Sometimes.
Can you zoom over one here? If you want here.
If that wasn't tethered.
Looks like pretty nice, but that's such a short leash on that.
Okay, got a hold of it again.
So now,
You can try down low there while you're at it. See what's.
sure. Zoom back in for you.
there. Okay, there it is. No, that's okay. I can read it. Okay, not very warm there.
What's it at?
Not high. 2.5.
2.5?
Yeah.
Okay.
Well, we could go for the spot up high where we got nearly, we did get 20.
Yeah, I think that would be better. Ideally 20 to 30 is what we're shooting for.
Okay.
Thought I saw a little
It's hard
shimmering.
because their legs make it look like it's shimmering.
I thought I saw a little shimmering here, but yeah, I can't tell.
Like their little legs move.
I think it might have just been moving around, but we could just put it in there and see if you want, you know?
I think we already tried that hole. Yeah. It's not very warm.
Alright, so you wanna go back to
So, yeah, more over on the right there, I guess, is where
the, sure.
it's a little warmer.
Coverage going up a slight bit in that area, 0.8.
Monica, was that the site that you were talking about?
I think, yeah.
Okay.
Yeah, there's not a lot of heat coming out of this. What do you think, Sabrina? Should we move and look for something else, or do you want to?
I mean, it has to be like a little bit higher temperature than 4C, I think.
Right. Yeah.
I mean, it looks like there's some flow, but it's not very hot.
Where are you at right now? It's one of those 6.
What if we just go straight back where you hit rock, or? Keep in there?
That's getting flat perspective.
Yes.
It's around here, you can't really pinpoint it. You know, it's
Well, we did have a spot in there that got up to 20 degrees.
Yeah.
It's really close to that.
Almost right up over here where they're all kind of.
Yeah.
Yeah, that's fun. Yesterday we had 20 and averaging 13, 14.
that a little flexible for you guys?
Yeah, we don't need to be on deck by 5. We only need to be on deck by like latest, I think 8 o'clock.
Okay, so then
Because the subs, the AUV is not going to be up till 9:15 or 9:30.
okay. All right, so we can stay down longer then.
Yeah. What do we need? 3 hours to get up, or 2.5?
Yeah. So schedule 3.
3 hours. Okay. So, all right. So then we got another 3 hours left if we.
That work for you?
Yeah, that should be enough. Okay. There's some rocks falling down in there.
Very excited. Wherever you need.
Oh boy.
It's not going up at all.
What about right under here, done before this already? Remember this little guy? Right here?
Okay. Well, maybe we need to move up.
Yeah, I'm ready to whenever you are.
Yeah. All right. Let's look a little bit more.
Would
This
we,
is not hot enough for that. 20 to 30 is ideal. 20 would be okay.
Doc, did we already check on the bottom? Just because I would think a lot of the rising would be what keeping these guys here, huh?
Yeah.
We already go down?
Did we? Well, we didn't make any temperature measurements down there, so we could look there. Yeah.
You want to do that?
All right.
Sure. You happy, Doug? Yeah.
That's all I'm using.
Okay.
There's somewhere.
All right, come on in.
Quite a bit in there.
Stable-ish.
Okay. Let's see what we have down here.
Those that they're kind of moving more down here. Oh, I'm pretty sure it's a camera there. Right where you are.
Oh, yeah.
The orange is right in that little darker there.
Oh, yes, starting around.
Look at that shit coming from below. Not going out much.
All right.
I see it. It's shimmer right there.
Yeah.
Almost under us.
Yeah.
All right, can we go down a little more?
Yeah. Right.
Sure. All right. Your arm safe?
Yep.
Yeah, that's not really diffused though. That's just straight up smoke, huh?
Yeah, we got smoke in there.
Well, we can see.
Sorry, Jim.
You can see it now.
Take the shrimp blower.
Look, there's a fish in there.
Yeah, there's a fish going through there.
All right, we're gonna go up top a little bit. Just we'll see here. It's gonna run with me.
Shrimp hurricane.
You good song?
Says the story of
Band.
the shrimp hurricane. The day the ROV came.
Those towers in the back, I don't look over too. That's great.
Shrimp and
See, that's right where we look. It doesn't look like it, but that's where we were looking before. It's right where it was dark, man.
Well, we were up there already.
We can park a little higher than we were before, and we'll try poking around a couple up here. Off this rock kind of thing. You wanna try that? Or you wanna keep flying around though?
Oh. Is there?
There's definitely flow beneath us, but it looked almost
Yeah.
like black, you know, like I don't think that's what you want, is it?
That might be a little too hot for them. Yeah. So, oh boy, I can't tell.
I'll park right here and we can poke around a little.
I mean, we'd settle for a little bit hotter as long as it doesn't, I mean, I feel like that would be okay. Anywhere like really below like 50 would be okay.
Okay.
All of our other samples have been 20 to 30, but a little bit hotter would be okay.
Okay, let's go for that smoky stuff. See what we get in there.
Sure. All right, come back down.
Alright, Joe Gravino is gone.
Good night, Joe.
Another amazing dive.
Yeah. Thanks for hanging out, Joe.
Shrimp. John, you want to take over? Are you good for the night now? So, no. That's where it goes to see. Standard. Go with the number 2, all lowercase.
All right. got hot water here.
You could log in as you.
It's not hot, I'll even really shrimp.
Let's see what we got in there.
How's the rock crab?
So you go right down in the middle of the
Proceed as
daily ball of
in
shrimp.
the morning. There's gonna be a big thing. Oh, yeah, I'll give it to her.
And 11, 12,
I wanna see this thing. They're broken into like 2 or 3 giant
12.
They put the basket out.
Right at the bottom of where the shrimp is.
The basket. Open up the bio box.
Going down.
They just
And so,
I think it's like 2 or 3 pieces. It's probably
and so 13,
4 feet long.
14,
Yeah, that's a lot worth it.
15.
Yeah. There was a okay.
You want to do here?
Yeah, here would be nice.
All right. So, 1, shrimp water.
Fluid sampler.
What you need, Chris?
Oh, my sister's on there. Hi, Jenny.
Okay. Sure. Rocking it.
I was working on that.
Hey, Jenny. Yeah, late night here for sure. It's 10 after 2 in the morning. We are trying to get a sample of warm water in the shrimp for some microbiology experiments.
I can forge out.
Stable isotope probing experiment. Culturing with C13. Seeing that get incorporated into the DNA.
No.
Got that gas tank. Yeah.
I might have to get Kevin to run this thing. Okay, we're at 3,745 meters deep of Mid-Atlantic Ridge. Area we're calling Cluster 2, Grappe Deux in French. It's a site where Ephemer has been doing some research. They've got a bunch of exploration areas in this general area of the Mid-Atlantic Ridge. And we've been here for, let's see, 4 days. And we've found this hydrothermal vent site. Found it using a combination of water column surveys, looking at signals for hydrothermal plumes, and using the autonomous underwater vehicle to map in high resolution to the point where we could actually see these structures on the seafloor and pick out several of them to go and look at. And we found a few that were extinct along a transect, and eventually when we looked in the right depth region, we found this one that's producing a lot of smoke. Spread out over about 20 meters of depth on a steep slope. Generally, it's volcanic rock that we're seeing on the seafloor here, pillow basalts. But we think that there's mantle rocks right underneath this layer of volcanics, reacting with the hot water and producing really large amounts of hydrogen, dissolved hydrogen in the water. We're trying to get a series of chemistry and microbiology samples here now. This dive is just set up to do sampling at the smoker vents. And right now we're trying to get some lower temperature water where microbes are growing. Just trying to get set up with the fluid sampler. So looking at the intake nozzle there, that's titanium. It's got a temperature sensor inside of it. And we can put it right down in there amongst the shrimp, and we'll pump some warm water through it and see what kind of temperatures we get. We've done a bunch of sampling already. We're up to, next sample will be sample number 18 for this dive. About 10 of those are water samples. 4 of them are biology samples and 4 of them are rock samples. We got a large piece of the sulfide chimney, almost a meter long when it fell onto the basket, and we broke it into several pieces and put it into the big bio box to bring it back up. Wouldn't be surprised if it weighs close to 100 pounds, the whole thing. I'm trying to get my pump running here. Okay, not happy with that. Try again.
What's the temperature?
Waiting to get the pump going. It's currently 4.8 with no pump. And all right, so I'm just getting about 4.5 degrees there. You can just move around. I'll tell you if it's going up or down. Oh, that's going up very slowly. 5 degrees, 6 degrees. 6.5. That's going up. There we go. 8. Still going up. 10 degrees. 12. 14. 15. 16. Okay, that looks like a good spot. 18.
Are you doing the cylinder or the filters first?
Whatever you would like.
How about the cylinder?
Okay. We're up to 20 degrees, 20.5. I think we can look around. So is that locked off and the arm locked off? Or?
Yeah.
Okay. All right, so we're going to start taking a large volume sample, which takes a while.
Sabrina, are you logging there? Are you logging the fruits and electrolytes? I can, but I don't. I can do it.
So it'll be S0498, HFS 8. We're on 18, right?
18.
Okay.
Yeah, is it 18? The next one? 18, okay, thanks.
I've been calling these large volume, but volume.
I also need LV.
Okay, we'll do that. Okay. All right, depth is 3745. And the heading is 80. And this is shrimp. Okay, it's hanging in there about 20 degrees. We got up to maximum 21, I think. And we're gonna go for 3 liters of water here. All right, starting a sample. Okay, that did not start.
Got that flood, this is with, stuff, so
I'll try again. Okay, that one is starting, and I see a lot of shimmering water coming out of the sample pump. So we are definitely pulling water in. And that's just going to run for about 15 minutes. So I could put Sabrina on. She could tell you what her experiment is about and how it works. I'll just say we've been out here for 26 days, and we've studied 3 sites. At each one of those places, we found new hydrothermal vent sites, like the one you're looking at here. So that's a pretty good record. We were looking for a different type of venting, a low temperature venting of high pH fluids that make carbonate deposits on the seafloor, but we haven't found them. We have found some very interesting chemistry and looking at the geology of oceanic core complexes where detachment faults form, where the volcanic layer is removed from the underlying mantle. The mantle comes up and gets exposed in places at the seafloor. Those minerals in the mantle rock react with seawater at a range of different temperatures depending on what depth they're at and whether there's a magmatic heat source or just the heat source of the mantle itself from upwelling. And this particular site here seems to have extremely high hydrogen content, which is consistent with seawater or hot water, hot reactive water reacting with mantle rocks. Could be fairly close to the seafloor, so just underneath the volcanic rocks that we're seeing at this site. We know there's been some mantle rocks that are sampled about half a kilometer from here at a little bit shallower depth, and the theory is that the mantle is coming up here and being uncovered. And there's just a thin layer of volcanic rock on top of it at this spot, which leads to the reaction that generates these really large amounts of hydrogen. So these shrimp and the other animals here have chemosynthetic symbionts that provide nutrition for them. They are a symbiosis, and they don't actually eat things they don't chew and swallow things. They have an internal colony of microbes that use the chemical energy from the water to produce organic material that feeds the shrimp. So the shrimp carry the microbes into the water that has the energetic compounds they need, like hydrogen sulfide and potentially also hydrogen. Generally, hydrogen sulfide is the main energy source, but we're not sure. These shrimp may also be using hydrogen because there's so much of it here and there's not a lot of sulfide. So we are taking a sample of 3 liters of water for some experiments on the ship. We'll bring that up in a few hours. We've got another site nearby that we want to go to take some more hot water samples. So that'll be coming up in a little bit. Okay, we've got a maximum of 25 degrees so far and an average of 22. Temperature fluctuating a bit.
This whole area is shimmering. Every time I zoom in, you can see a new shimmer.
Mm-hmm.
So just looking at one video about so some of the shrimp down on the arm. On the arm.
Yeah.
And I don't know if you want to see it or not, but one of them was holding what looked like a piece of another shrimp, and it looked like it was eating.
Oh, really?
Yeah, it was going like this.
Okay. What's all over? You got snails all over the arm?
The orange is torque stripe, which tells us that the vulture.
Oh, that's just your torque. Okay. Huh. Usually, is it actually red or is it
orange?
It's orange. Okay.
But that one doesn't have it.
Okay. Yeah.
That's right.
Yeah, it's just like that.
Okay. Yeah.
Oh, look, we got friends again today.
Shrimp climbing on the arm. Well, we don't have a totally full house in the room tonight. We got 10 people in here.
See that?
Yeah. See how shimmery it is? It was not like this on the other side. I don't know if it was the current or what, but
Yeah.
everywhere here.
Our temperature's dropping a bit. I don't know if we need to.
We haven't moved, but we can move if you want.
Down to 9 degrees. You might want to try to move it around a little bit, get it to come back up.
Sure.
Move around in there?
Yeah. That's going up. Yeah.
You thinking about Niskins here or the other?
We could do Niskins here. We just can't do them at the same time as we're doing this. Yeah.
You doing that?
10. Sort of stable.
Alright.
You want us to keep moving it?
Try to move it a little more. Let's see if we can get it back up towards 15 to 20. That's going down a little bit.
It's that red.
That's going down. Yeah.
Moving down?
That's going down.
Going down.
Coming back up.
10, 11, 12, 14, 15. You'd be good with that one?
Yeah, and it's still going up. So
Anybody want any starters?
no, thank you. Okay, we're almost to the halfway point for this one. Alright, so we got 37:45. I already wrote that down. Alright. Oh, now it's falling down again. You can move it around, just it's okay to put it all the way up against whatever the surface is in there. Drop down to 8 degrees or
I think we're getting a little bit far away, so whenever the current shifts, it's coming out of it, you know?
yeah.
Coming up. 11, 12, 14, 15.
Yeah, there you go.
Yeah, that's nice there.
That's better. Okay, now we're back up to 24.
I'll look at our gauges real quick, Doc.
Yeah.
Terms of 5. So Sigma is actually the highest on all of them. Pressure-wise. So it really shouldn't be going from turn to Sigma right now. It's weird.
What's going on with your gauges?
So that one that says sub M
Yeah.
is actually going up very slightly,
Mm.
which is very, very weird. I don't want to see that. There's smoke coming through. There's smoke coming through. That's it.
Quiet in here. It's late night.
Early morning for us, yeah.
The biologists were very happy a little while ago after taking all the samples that they got. They got lots of different types of animals, plenty of things to work on in the lab when it come up.
Like they got some scale worms or something, and some pearl stars, all kinds of stuff.
Scale worms, different types of gastropods, yeah, or polychaetes of some kind.
We need to come up for an AUV win.
Oh, we need to come up, need to be on deck by 0800. They're not going to be up till 9:30, but 5 o'clock off the bottom.
We got a while. Did y'all explore around this area any, or mostly just come right here?
We didn't do much exploring. This is mostly just for sampling, but we took a rock on the way up to this vent site, and we'll try to move around to that spot that we sampled on the first time here. Once we get through these.
Oh, the north side of it?
Yeah, the uphill northern side of it.
I'm sure you saw over here too, there's a big, it's like a crown with shrimp all around it. And then there might be some over here too. I don't think we went over here.
Yeah, we want to go back and sample that same hot water vent that we did the other day.
It's pretty good. Every now and then it blows, current kind of blows some hot water through the shrimp. You can see them move like in a wave.
Yeah. These shrimp have certainly figured out a way to multiply. There's also a lot of the snails at this site, and they are scavengers, and they probably are consuming the shrimp, the dead shrimp.
It's kind of crazy to think, like, how did all this start, you know, like when this vat first formed. And then these shrimp, I guess, just go out and kind of wander and look for these, or?
Well, the theory is that the shrimp produce larvae that float through the water, and if they happen to come across a vent site, they settle there. And once they get going, there's so much larvae in the water all the time, they just keep on recruiting new shrimp, new juveniles.
So the larvae don't need to be at the vent to survive.
The larvae can float around in the water. I don't personally know very much about shrimp larvae, but in general, larvae can float through the water for sometimes pretty long periods of time until they happen to come across the right habitat where they can live. The currents along the ridge axis where the most of the vents are found, are sometimes directed along the axial valley, so that if these animals produce larvae, they're going to float up and down the ridge axis. And the natural flow of the currents will probably get them in near another vent site, so they can spread from one side to the next. Oh, we're done.
What was the maximum temperature?
Maximum was 28.6.
28.
Yep.
Wow. And the
The
average?
average was 19.
Yeah. And the volume?
3,000.
Thank you.
Okay,
Oh.
so we have Falkors 14 and 15 on.
Okay.
So those two, and then that's it. 3 liters each.
Okay, moving to number 14. Okay. Starting another sample now.
It's filter 14, correct?
Yes.
Yeah. Thanks.
Time on this one. Oh, that's UTC 0539. Got little spots on them, little speckles. Okay, Sabrina, you want to come and talk to the people about your experiment? Because I can't.
Okay, yeah.
Can't even.
They want to hear what it's about.
Okay.
At least somebody out there is listening. I'm mostly thinking about going to sleep.
I know, me too, honestly. Okay. Hi everyone, my name is Sabrina Alcasis. I'm a third-year PhD student in the MIT Woods Hole Oceanographic Institution Joint Program in Chemical Oceanography. but my research mainly focuses on geomicrobiology. So right now we are taking a 4-liter cylinder of water from these sort of diffuse sites, so they're not focused black smoke or flow, which is too hot for microbial life. Instead, we sample from these zones of mixing, so it's hydrothermal fluid mixed with seawater, and they're very productive zones for looking at the microbiology at hydrothermal systems. So usually we look for temperatures between 2C and 120C. But right now we're in a really good sweet spot at, let's see, 15 right now. So what we're using the water for is 2 separate sets of experiments. The first is a large suite of different culture media, and we're testing what can really grow here. Originally, we had made a lot of different culture media that had high pH because we were hoping to find a sort of lost city-like system. But we also brought sort of some standard medium for heterotrophs, and then also some other autotrophs, some nitrate reducers, and sulfur oxidizers, as well as methanogens. And so in total there are about 7 different media types that we're testing at 30, 55, and 80 degrees. And so once I get back to the lab at Woods Hole, I'll be trying to transfer those cultures and then seeing if we have any new isolates, potentially new species of microbe from these 3 really awesome vent sites. The second experiment that we're doing is called RNA stable isotope probing. And so for this experiment, it's a pretty complicated experiment, so I'll try to break it down. So first, we take the fluid from this 4-liter cylinder, and in 1 bottle, we empty it into 4 bottles to test 2 different temperatures. So we're doing 55 and 80 C, but we pair the experiments. So at 55 C, we have 1 experiment that we add an isotope to. So we add 13 carbon labeled bicarbonate, and then in the other we add just regular 12C labeled bicarbonate. And then we add hydrogen to the headspace. So ideally we're targeting autotrophs, specifically chemolithoautotrophs, which are microbes that use the chemicals from these systems to produce carbon, and that's what supports these very diverse and rich ecosystems, and that's actually how these shrimp are surviving. So once we add this isotope labeled carbon source, the bicarbonate, then the hydrogen, which is their chemical source, we incubate them at 55 and 80 C, and then we filter all of that. So the point is that the active autotrophs, or the microbes that are actively metabolizing, will incorporate this isotope, this labeled carbon, into their RNA. There's also DNA stable isotope probing that my lab also does, but specifically I'm interested in the RNA because what we can do with the RNA is figure out what genes are actively transcribed. So not only can we figure out from this process who is there in this autotrophic community and who is actively metabolizing, but we can also figure out what their specific functions are and what genes are present. The experiment at sea is relatively simple. We just add the fluid to these bottles and then add the isotope and then incubate and filter. But then once we get back to the lab, it becomes a very intense molecular biological process where we extract the RNA and then we add some things called DNase, which we try to eliminate any DNA in the sample. And then in order to separate the heavy RNA, or the RNA of the autotrophs that took up this labeled bicarbonate, from the inactive autotrophs, which didn't take up the label, we do what's called a density gradient fractionation, where we have a density gradient solution, we add the RNA to it, and then we basically spin the heck out of it, so 65 hours at 37,000 times gravity, and that's enough to let the heavy RNA sink to the bottom, and then the lighter RNA, which again represents the inactive autotrophs, that will stay right on the top. And then eventually, through more experimental steps, we then can sequence the two fractions and look at who was active and who was inactive, and then process it using bioinformatics. So looking at we call it the RNA stable isotope probing metatranscriptome, so looking at the genes that are transcribed and also pairing it with these two filters that we're doing now, which represents the unmanipulated diffuse fluid community. So yeah, those are the two experiments that we're doing with this fluid. We're also taking cell counts just to look at the general amount of cells in this fluid, which is very high usually compared to deep seawater, usually 5 to 10 times as abundant as in regular deep water. And then we're going to give some of the fluid to Dave to look at the fluid chemistry, trace elements, etc. And then we'll also give it to Tamara, our gas geochemist here, and she will give us readings on hydrogen and methane from these fluids. So yeah, that's it. Okay, one more liter.
3 and 5. Take off it's fine. So 3 and 5 is 1, 2, 4, 6. Is the bigger and somewhere on the left. Fire them all right here. And we've got sample, you can see what's left. Let me write this down.
Okay, so I see a question from Connor. Are you guys going to sample the microbes and the shrimp are harboring as well, or just as a comparison? So I'm not going to do that, but someone else from my lab is going to do that. We mainly just sample here because where the shrimp are clustering is usually where these diffuse sites are. And so Bailey Benner, Dr. Bailey Benner from my lab, collected a bunch of these shrimps from all of the sites we've visited so far, and she's going to do work on their viral symbionts, but she also will sequence their genome, and she extracts their lungs, which harbor chemosynthetic symbionts, and so she will get a good idea of what symbionts, microbial symbionts, are with the shrimp, as well as the bacteriophages or viruses that are affecting those infecting those chemosynthetic microbes. Let's see. Yeah, there's certainly a bit of a cluster, indeed. These are the most shrimp I've ever seen in my whole life in one place. Oh, sorry, lungs. Actually, funny story about that. We were so tired after the last dive that we had labeled all of our samples as lungs, and then at the end we looked at each other and realized that, yeah, obviously they don't have lungs, they're gills. But I think my brain is still not working and I'm still saying lungs. gills.
So that's what we have left for samples. 4 pistons. 5 bags.
Oh, thanks, Ryan. Yeah, I'll try to get some rest. Hopefully after these 2 filters, I can go have a quick nap before the ROV comes up. And yes, that can potentially identify novel metabolic pathways. Yes, it can. Are you talking about the stable isotope probing or the shrimp chemosynthetic symbiont experiment? Because both of them we're doing metatranscriptomics on, so if a pathway doesn't match up to what we find in the database, it's probably a novel pathway, and we usually investigate that more. And then also with the culturing, if we have something that doesn't match up in blast, then we potentially have a new microbe. I think we're at 2,600.
The next speaker was on which position?
14, 15 things.
All right, I'm gonna give us back to Dave, but thanks everyone for listening. We're almost done with the
Is it still
first
running?
filter.
Oh, yeah,
Yeah,
it's
it's
still
at
running.
2,700.
Yeah.
Thanks. I just went
On your computer?
no, I just didn't want to finish logging and then John can take over. So you can be in there. It's fine. I just had I started to log on this one, so to finish that. All right.
Oh yeah. I guess there's not much.
We got about 2 hours. Let's say, you go back down south, look for rock exposures.
Yeah, we can try to do that side.
Okay. You could go, just about any direction. I think you could go very far north. You might hit some sediments. But, if you go anywhere else,
I think I'm going off. There was a direction I wanted to go last time, but we ran out of time. I think maybe also chimneys. Maybe we just go there. Let's see.
sure, they're gonna do rock sampling out of that. If you found any rocks that, and John can help you with this, if you found any rocks that were not volcanic in here, that could be sampled, that you want to get, and not sulfide. We got plenty of sulfide. We got 2 volcanic pieces in here.
Already okay.
One of them,
glass on it. I mean, it's kind of dull, but it looked like glass. I think that's
Oh, it stopped, yeah.
Okay. Oh dear.
Not logging in.
Okay, maximum temperature was 28.9. The average was 18.9. And the filter volume was 3003. Okay. You're welcome. Then I'm gonna set up for the next RNA filter. That's 15. Yeah. And that's also 3,000 for the pump limit?
Yep.
Yep. I can, I think, log here. After 15 minutes. Okay, the temperature is around 10 degrees right now, so it dropped a little bit. Ah, it's rising up again. So I'm starting that one.
They can move it around if you want to.
Okay,
It's
I think it's
overall,
rising
it just
steep
goes
up.
to
So,
there.
yeah.
It does go up and down. So it's,
Yeah. I had it yesterday also.
yeah.
Yeah.
All right. So goal is to like fill it all up if you can.
Take as many as I can, yeah.
And at least once you get set up there, the chemistry samples go pretty fast.
Mhm. But it's probably also good.
It's a lot.
8, half an hour, actually more, 45 minutes maybe.
Yeah.
It's gonna be close.
About 5 minutes each.
Yeah.
Sometimes less. But,
Okay.
and we've been doing like in the bags only 350.
Oh yeah, that's right. 350. I should
just for these.
not
specific
4.
gases.
Okay.
And pistons could be 500.
Okay.
Just if you want to go to the, you're talking about going to the north side of the fan looking south.
Yeah.
Probably have to do it. That would take too long.
Okay.
Oh,
Just pop right over.
I see. Okay.
Get our umbilical off from
Right.
Okay.
It's not right over
So
there.
it's other direction is better than
That
that.
would be the next move after this. And, if you fly through this, so gonna fire some Niskins here. Tomorrow's got that all.
Fire them.
Niskins. You.
Can
yeah,
we
so
fire
you
them
can't-
while we do that sampling, or are we gonna move things
no,
around?
no,
It's
no,
gonna
no,
be
no.
down, right? It's still, yeah.
But you can't fire them right after you put this away.
Mhm.
Yeah, so we are doing this filter, which takes probably another 10 or 15 minutes, and then I'm gonna take one more unfiltered bag with the HFS, and then we're gonna do the Niskins and all on this side, and after that we're gonna move to the hot temperature from yesterday, like what is it, about 10 meters above, right? And Dave gave the depth and the heading we had yesterday.
Yeah, pretty sure I remember.
You remember? Okay, good. I only half remember.
Okay. I'm leaving this in your hands.
Okay.
Thank you.
Filling it all up.
Yeah, if you need help with that,
I'll find Kevin.
Kevin's in there.
Yeah, that's great. Thanks.
Hopefully it'll be an easy setup there.
I hope so.
It was a little tricky the last time,
Yeah.
but you can get it.
So shall we first go? We know the high temperature. I guess we can go straight with the same. We come on with another HT probably before we start. Just to know.
Probably good to get.
We get the same thing.
Yeah. And hopefully this will be close, although. Yeah, it's probably good to get another IT.
Mm-hmm.
Yeah. All right. Good luck.
Thanks.
You want one of these?
No, I'm good, thanks. Tons of chocolate already. That's sample number 20, right? The second field cut. Yes, thank you. So we are still sampling for microbiology. We are about 1/3 through the sampling volume. So we are right now filtering 3,000 milliliters of water through a filter, and after this one we're gonna take one more bag sample for chemistry, and then we will sample for Niskin bottles, and after that we're going to a new position with some hotter fluid with black smoke, like the same place we were yesterday, to get more chemistry samples. So Sabrina's just been talking about her experiment, and for the chemistry we are going to look at the fluids but also at the dissolved gases. So we will measure some of the gas here on board, and then also do more measurements onshore to get the full composition of the dissolved gas in these type of samples, and also the fluid or the liquid part of the sample, like what the water composition is. But most of it will be onshore analysis. Here on board we do measure the pH, and we do measure the hydrogen sulfide content, and some of them we also look at the silica content in these type of samples. I wonder how many layers of shrimp it is.
It's crazy.
Yeah.
And there's a lot on
At times some of them are just like several layers.
There's a lot on the other side as well.
Yeah. Yeah, we are up at 20 degrees Celsius again, so good temperature.
So I think the most diffuse thing I've seen is tray.
So far, yeah. It's very intense here, yeah.
There's nowhere I can zoom in that's not shallow.
Right now we are at 1650, about halfway through currently. Yep.
Some of these would have heavy metals and stuff on them too.
You know, probably yes. I would think so.
With the arsenic sauce. been trying to zoom, and every direction I try and zoom, there's shimmer.
Yes, it's
There's
crazy.
nowhere I
Yeah.
can just zoom in to the closest up front of the arm. There's so much heat.
We crossed the 2000, so yeah, getting tired. This is amazing.
Really, really cool too.
Mm.
I wish I could bring one up and keep them as pet.
Well, there will be plenty probably in the box that came along with the chimney.
That would make them frightened. There's a lot of times tonight if you look at the table, it has a brick, or it has a home. Or it has a universe. The last time they run together with the but when we begin first though, see look here, see the first line like this. Right? First line here. Somewhere a little later. And you have the first half of the ups and downs and downs and downs. Very half of the ups and downs and downs. You know what I'm saying? And a little movement there will be a lot. But that's what we do. So it could be the term, but there's just not how much about that theory, like what is the constant pressure gradient? Right. There's actually more pressure for the gauges in the same. In terms of why is it? Well, that's probably the subject. If the output can actually
Can we check these guys there on the corner, the trees that are more orange?
The ones more outside, the orange guy.
Yeah, the corner in the other corner.
On the left side, yeah.
Yeah, those more.
That guy's high in the orgy, so. Weird.
Those are two species.
Are we looking at two different species here? Or? Okay, Sabrina, it's done. Yes. Okay. The maximum temperature was 28.8. Then the average temperature was 24.1. And we filtered 3001 milliliters. You are welcome. Okay, I'm setting up for one more chemistry bag with the HFS, and then we move on to the knee skins.
We got a bio question.
I think we need help from Joanne for biology. So that's 21, huh? HFS 21, and the last one was expect 21, and also sample 21. Is that correct? The upcoming one?
Yeah, it's 21, but is it RNA 16?
No, it's going to be back 21. Too many 21s. You're welcome. Good night.
Oh, these are fish.
Now we finished sampling for microbiology, and now we switched over to our chemistry bag. So this is only going to take a few minutes.
After this is missions.
Yeah, 4 of them. We're gonna fire 1, 2, 4, and 6 at this very spot here. Yeah. The 3 and 5s are coming when we leave the other side in the black smoke. I think we're not entirely sure which one are intact because
We already did both handouts.
we took both of them at the same spot. Yeah. I have one gas site left that I'm gonna take in the black smoke or in the next setup. Okay. Yeah, nobody left. Okay. Okay, the chemistry sample is done. So we can store the wand and then take the 4 Niskin bottles at the very same position that we are at right now.
I got a shrimp on the camera.
Well, there's everywhere.
Where you going, buddy? I'm gonna need to torch out so I'm going to here.
I don't know any schemes here.
Yeah.
Shelton to fire 2, 4, and 6 together.
1, 2, 4, and 6, yes,
Yeah,
that's
okay,
correct.
perfect.
And then,
3 and 5.
yes.
Perfect.
Yeah.
Okay. Well, you let me know when.
Yeah, so we are now setting up to do the first 4. These
Okay.
are the 1, 2, and 1, 2, 4, 6 is coming up now. Yeah.
So there's a significant amount of shrimp in our biobox.
Yeah, I think they came along with the chimney. So that I don't know if you were already awake at that point, but we sampled the big chimney, and there was a lot of shrimp sitting on the chimney, and for a while there was still fluid like coming out of the chimney, so they kept eating while we basically put it in the biobox. So I think all that shrimp came along with the chimney sample. Yeah.
The lower lights are on. Yeah, it's just smoky everywhere. I can try to turn it upside down off if you want. Yeah, all lights are on except for the floodlights. Look, you got a little bit of shrimp shell on there too. Yeah.
Oh yeah, good morning. My name is Tamara Baumberger. I'm from the Oregon State University and also a NOAA PML program. From the Earth Ocean Interactions program. I'm a geochemist, a bit focused on gas chemistries. I'm the head of the helium isotope lab in Newport, Oregon, and I'm also doing a lot of work with methane, hydrogen, carbon, basically carbon and hydrogen cycles, including concentration and isotope measurements. So on this cruise here, I'm responsible for all the gas tide sampling, and I'm helping out with fluid sampling right now. We also have a high vacuum extraction line on board where we can subsample all the gas tides, so we can split up the fluid into gas phase and then seal off the gas phase in some glass ampules and take them home, analyze them for helium in our helium isotope lab, but also analyze them for all the other gases at the University of Washington and at the ETH Zurich in Switzerland. So right now I'm doing all the fluid sampling. Our chief scientist, Dave Butterfield, went to bed and he gave me some instructions of what samples he would like to collect for the rest of the dive, and I also want to get more gas tide samples. So we just finished up sampling for microbiology and chemistry at this diffuse vent. So this vent is kind of a mix between the high temperature member and seawater. It's a subsea floor mix already, so the water is already cooled down. So we were measuring between 20 and 30 degrees Celsius during our sampling time. And you can see that the shrimp think it's quite cozy here, so we have a lot of shrimp around. They feed on the vent fluid that is coming out of the seawater, or coming out of the seafloor, sorry. And so now we are setting up to take some Niskin samples. So these are for eDNA, for metals, and also gas analysis. We will subsample them tomorrow and also run some of the samples on board here. And after that, we will move over to the spot we have been sampling last night. That's going to be a high temperature fluid. We have been measuring over 300 degrees Celsius when we were there on the last dive. So we want to pick up a few more samples because we only got one for fluid and 2 gas tides. So we have like a bit of a sampling list left to get us a little bit bigger diversity for samples. Typically when we do the gas chemistry, that is giving us a lot of information with what type of rock the circulating fluid interacted on its way, and also on what type of temperatures, like how hot it actually got, how close it could be, or if it's just the fluid venting is influenced by hot magma source, like with lots of magmatic gas, lots of helium and high heat, or if it's more like something a bit shallower that doesn't have a huge heat source, like when we do the serpentinite hosted systems with lower temperatures. And so with doing all the gas chemistry, we kind of can track down with what type of rock the fluid interacted, and that also tells us what tectonical settings we have, what input, a whole bunch. So we're gonna fire Niskin 1, 2, 4, and 6. Okay, I have to lock the Niskin bottles. That's going to be 22, right? The overall sample number. I think they want them all together, like, yeah.
22?
Mm-hmm. And then N1, N2, N4, and N6.
So we should put that all under 22?
Yeah.
What's that? Yeah. And then for the time, do we use the current time that we have in the cell phone?
UTC, it's 6:32 right now. Yeah, 1246.
Look at those little fancy flags popping up.
Yeah, it's pretty fancy now, yes. The altitude is 1 meter. Shall I have a look? This is great with the numbers.
Yeah. See, 1 fired, 2 fired. What was it, 1, 2, 4, and 6?
Yeah.
6 fired.
Yeah, 4 is always tricky to see. Yeah.
How you would define the
I
area?
can move the arm out.
I wrote broken spire, diffuse, flow free.
Okay.
I don't think it's like that. All right, it looks like we got them. Huh?
We haven't verified 4 yet.
4, yeah, okay.
Yeah, go for it now.
It's right in the camera.
I think we just have to trust Carl's flag on that one.
All right, I'll submit the sample, so it's in.
You shouldn't be able to move if it doesn't go.
Yeah, okay. Well, that's great. Okay, that was it for this diffuse flow site. We have taken all the samples here, and now we're gonna move back to where we have been yesterday with the black smoke. It's kind of where the shrimp. We left the physical marker, the seafloor marker, at the shrimp spot, and the high temperature spot should be about 2 meters lower than the shrimp spot, I think. And I know, yeah, that you know. I don't, so I'm glad you remember properly.
Pretty sure.
For me, it's a bit of a guessing game.
Do we want to have a quick look around and an overview of this area before we go over?
That would be nice, yeah, just kind of taking a step back and take a few photographs.
I think we're going to have to move the boat anyway, so.
All right.
If you guys remember yesterday, where we ended up being was, so if you follow this up with the shrimp, on the other side, it kind of spills back down the other side a little bit, but not quite as much. And then kind of down and to the right from where I am is where that hot vent sample was. So we're gonna have to be a little more than 180 put that way. So probably we're gonna need to get the boat, like I'll come off of here, I'll back up, I'll come around this. This is another one. So I'll come around here, I'll look at it, get the boat up at least over here, I would think. All right, can. All right.
So we are going to reposition to sample high temperature vents, and both the ROV and the ship has to reposition to do that. So it's going to take a little bit of time until we are where we want to go, and our next sweeter sample is going to start. Yeah. So before we leave, we're going to have a bit of an overview look of the whole area here, taking some video. Someone is asking if I've been on the EV Nautilus. Yes, I have sailed on the Nautilus three times, I think, yes. And all these cruises have been methane seep related at the Cascadian margin.
ROV Doug speaking. Okay. We just put a new waypoint up there. If you can start making your way up there, please. Thank you. Bye.
Mm-hmm.
On the other side is where we will be big.
Mm-hmm. They're quite tall spirals. And I've never sailed on the Oceanus Explorer. However, we have been guiding dives from ashore. So that means we had our little dive center set up in Newport, Oregon, and we're directing the ROV dives from shore while the people on the Oceanus Explorer helped us getting all the samples we wanted and the right video images. So the cruise was based on some watercolumn work we did on the Falkor in 2015, and the Oceanus Explorer went there after we found some position of new hydrothermal vents, but at that point we only had the water column like CTD and AUV with us. So the Oceanus Explorer then dove on targets that we found from CTD and AUV. And after that, the Falkor also went there in 2016 to collect more samples.
Should be well above them. 10 meters higher than the highest one on land. It's always shrunk off.
Yeah, this is my 4th time on the Falcon now, and I've done 3 times Nautilus.
Start heading down, looking for plan. Forward a little bit. Starting to see the ground. I need to check out this thing as we go around.
Mm-hmm. Sounds good.
Ground. So there should be a smoker right in front of me. There it is. That's the one that looks like a castle. That's the one I was talking about. That's what was smoking us out when we were flying over this. What's that? So this is not the one we were sampling?
Yeah, this one has like this very diffuse kind of like smoke, not very strong.
Different one. So I'm going to wait here as the boat comes around.
Hello, Eric. We made our waypoint a little too close to us. Can you just bring it to the west about 20 meters? As usual,
Yeah.
Thank you.
Yeah, I never turn them off. You do.
Wow.
Yeah, this is a cool one.
Mm-hmm.
We just came by this briefly the other day. Shrimpopolis.
Yes, as well. Shrimp density is definitely high in this region.
A whole lot of those eggs again.
Mm-hmm.
See all the eggs?
Mm-hmm.
I gotta wait a minute. Always gotta catch up. I'll carry on to the left once they do.
Mm-hmm.
Just kind of look at this and then
So we are waiting for the ship to catch up, or the ship to get to the position where we want it at, and then we will move towards the site that we're gonna sample. While we are waiting, we're looking at this black smoker here. Lots of shrimp again, feeding off the diffuse flow.
Can we focus just a second?
where you want to? On the
Pretty much straight ahead of you.
Yeah, just in the center.
Just to the left.
So we're zooming on some of the eggs again, yeah.
Oh, okay. Yeah, perfect. Thank
Or
you
just
very much.
overall zoom. Yeah.
Beautiful. Wow.
Okay.
Thank you very much. Yeah, sorry.
All right, backing up. Starting to go around the north a little more. They're gonna move in a little more north, right?
Yeah. Where does he set up?
I think that's gonna be where our launch recovery is over the top. Yeah, I'm gonna have to kind of go around this thing to the, gonna be facing almost a little bit west. Finally get there. Creeping around some more now. Couple of shrimp on this one too. At least 5.
It's amazing. The whole, like the base is, well, this is, I remember that from yesterday also.
Molted shrimp.
Yeah. Yes.
It's almost like staghorn coral.
Wow.
It's a very impressive sight.
It's beautiful.
It's a crazy view.
That is beautiful. Just beautiful.
Another world down there.
Yeah, it is.
I want tomorrow to say something poetic here.
Oh, it's well on. All right. Moving on.
Mm-hmm.
You're gonna move them a little bit to the northeast now, right? Yeah, if you don't mind. I want them to end up being like just basically due north of where we are, but where I'm not in the danger zone.
So we are moving on again. The ship is close to its, or getting closer to its position. So we have a bigger range now to move.
Updated the launch recovery too. Yeah. Nice. Alright, so up here should be the site we work from the other side.
On that side.
Behind the other phone, maybe? Or, yeah, check the yes, there's our marker.
Mm-hmm. Yes. Yeah, charging. Welcome.
This is the back side of where we're going.
Oh yeah, now our seaflow marker is also coming up on the side that we deployed yesterday. Good job.
And our
high temperature spot is going to be the spires to the left in the distance.
Yeah, I can see them now, yeah.
So you want to swap it out with ours?
But now I got a little
Do you
more
have enough space?
to go more to the northeast.
Okay, perfect. Yes, yeah, we moved it. We'll try you on clear. All right. Thank you.
Where's your RV house check? Thank you, SOI.
So you see, this is kind of the heading I got going here after this, so that's why I keep moving them to the northeast.
What's that?
There's going to be a cliff to my right.
So now you see the black smokers coming up here. So this is where we're gonna set up for sampling again.
What are you doing? This is where we did our hot samples yesterday.
Right.
So they want to do another hot sample.
Yeah, lots actually.
The pistons weren't working right, I think, so we got one good one on the pistons and some gas tights.
That's right. We had two good both gas tights that we fired yesterday were fine, but the pistons didn't work, so we ended up with one good bag for fluid sampling. And yeah, we do need more. Mine
gas
went
tights
well.
work.
Yeah.
Days of this.
No good.
Why did we bother?
Because I'm nice and helping out.
A little more for both to catch up. No. This left is pretty rough, right? Shrunk a king inside his head.
Yeah, that's where we sampled in the.
Oh, I came in. Those things were all on my new shelf I built.
Oh, really?
Yeah. So I've got another place to stick snacks. So I immediately set mine up. As you plastic flamingos. Like the yard ones? Yeah. Matter. Tight spot.
So the heading yesterday apparently was 205, if that helps in any way.
Okay.
Yes, no, there's like they're in between each. Cliff and a chimney. Five. Can you do something with the lights or the camera or just the smoke doing that? Alright, I guess I'll go back up and we may not actually be able to get back to this spot if the smoke's blowing different.
Oh, okay. Oh, yeah. It's pretty intense.
I cannot see anything.
Yeah.
Over here. So we can try and park here and see if it clears up.
Mm-hmm.
You want? Or
We
we
could,
can look for a
yeah.
different spot.
Let's try and wait a bit and see if we see any improvement. And yeah.
I'm just gonna slowly
Yeah.
up in here. It doesn't have to be geoflat, right?
No, it doesn't. I just thought if it helps to find a good position.
Debris. Marine debris. It's up in the videos we watch.
Yeah. Okay, it's right in front of us now.
Is Blue View working or it's yeah?
Yes. Yeah, I see. Event history, I only see 1 and 2. 47.
Alright, we're there.
Yep.
This cooperates.
It's definitely right, the one right in front of us, right? Yeah. So now we just have to hope it cleans up a little.
Those solar photos
if you say try to connect, you can say second button on the bottom right. There you go. Yeah, that guy. Alrighty. And what are we going to do here?
So,
HFS one?
actually, the high temperature probe, your probe would be the first thing.
Find a hot spot and then take some samples from it.
Yep. If you can see at all. Yeah.
Looks like most of the shrimp.
Yeah, were they all fired at the same place?
Yep.
One of this is missing one. And then we'll allow this 1, 2, 4, 3.
Ranking it.
That's a lot to have collected in one spot.
Do you want me to torch out anything?
You're here for that?
That's all the way down.
That's it.
Yep.
The point for another
Tomorrow. I mean, yeah.
So we are now setting up to use the high temperature probe to get the temperature reading before we start sampling. So we're hoping that it clears up a little bit so that we can see a bit better.
So before they move,
We got a lot more torch out
tomorrow. The distance, you fire basically all in the same place at the same time?
there are 3 of them potentially don't work.
Got it.
So one of them, it's a whole story behind it.
Good, this, or bad.
So we should have one good one for sure.
Got it.
Wheel back.
So we're having 1 hour left at the seafloor, and I'm trying to sample as many fluid samples as I can in that time.
So they didn't quite come up for the long match.
This is the gas tank.
Oh, wow. What we gotta do now?
Yeah, well, I'm just trying to do it from the,
Yeah, the handle is in the way.
I think torch out, just, oh no, that one. Down.
That's it. Like just getting close to that smoke.
Alright.
Try torching in.
That's fine.
Nice.
Stop on the mark.
No, not yet, not yet. Here you go, the fruit, yeah.
Just trying to get it in the room.
Yeah, it's not. It's still hot at the bottom of it. Alright, what do you want to try first?
The one with the strongest smoke.
I say yesterday, or is this where all that smash stuff is? It's somewhere back in there, but it's really hard to see here. We have to really kind of feel it out. Remember, Doug? I think you were operating, weren't you? I wouldn't say you were in there.
I think we knocked off the chimney there. And, yeah.
Oh, this is where you guys got that big chimney sample today?
No, we knocked some off yesterday also.
Alright, so over here is where it's concentrated smoke.
Mm-hmm. It looks like chimney here.
Ten, 11, 14, 16.
There.
Yeah, we can try there, and if that's no good, we can go a little higher up there where the spires are.
Bottoming it out right there.
46, 50.
About
So we got over 300 degrees yesterday. So.
Yeah, I think you're at maybe like kind of where you were before, but back more.
Yeah, I wonder if it's like on top of these spires, like up on the upper, yeah, like there were kind of very strong smokes coming out.
Oh, right there? Yeah, yeah, right there. Right there, what the lasers are there, Chris? Would you be able to, do you think it would help if you came back towards me and came in more like this instead of like this? You know what I mean? On this tower. The smoke is strong.
Yeah, there were the smoke kind of, yeah.
There's strong smoke right there.
Mm-hmm. Can try it there. 10.
70, 90, 100. Yeah,
Okay, 130, 160, 200. Wow, 250, that's good. 280, 290. Yeah, I want to see a 3 there. 300.
You happy with that?
No, that's a good spot, yeah.
That's a good spot?
Yeah, let's do it here. Does it seem okay for you guys also with the wand and everything? Yeah, excellent. Let's do that. 301.
So now we'll go for the wand?
Yep.
Alright, back up.
Yeah, then once the wand is placed, we're gonna take 9 samples with the HFS plus a gas type that is also connected to the HFS. So our whole sampling will be the one position.
So get
Once we are set, yeah, get comfortable, and once we are set, we can take them on.
Watch out or anything, or something else. That's the next few triggers. Watch out. Yeah. Now you want to watch out, right?
Now you can watch out, yeah.
Right.
Let's do one.
There we go. Yeah, I'm not a big fan of this current. I can drive a little more, I think, if you want. All right. All the way.
Out of the black of this.
Yeah, just wait till we can see. Try pushing in, see if you're free.
I don't think I am yet.
So we measured 300 degrees Celsius with the high temperature probe in the chimney here, and so we decided that it's a good spot for doing the fluid sampling to repeat samples from yesterday. So now we are setting up with our hydrothermal fluid sampling inlet using this wand, and it has a temperature sensor also, and I have the controlling system here of the fluid sampler, so I'll be able to switch to the different pistons and bags to fill them up and control the speed of how quick they fill up and how much volume we're gonna sample, and I will have the temperature readouts at the same time. And I also have one of my gas tight samplers connected to this inlet system, so we will be able to just sit in one position and collect all the remaining fluids. And once we are done here, we will take 2 Niskin bottles in the black smoke above this. And I have a pretty decent view.
There's a lot of smoke to your right, so if you can kind of come in a little like this angle instead of this angle, keep your arm further away from that. You know what I mean? Yeah, like that.
Okay, temperature probe is rising. I'm at 30 degrees now, 40, 50, 60, 70, 80, 90.
We established this probe working at the same level of our hydro probe, or is it?
We did calibrate it, but I think Dr. Butterfield still trusts ours more.
Yeah, that's why he wanted to have a first reading with yours. We are at 110 right now. I'm not quite sure. Actually, what temperature? He had 200 degrees on the. Yeah, he read 260, while you guys had almost 300 degrees at the other end. Okay. So we should be able to get a little higher actually here. Yeah, I'm at 90 degrees right now, so I do expect that it would read a bit higher in a perfect setting. Still 90, yeah. It's rising. 100. 110. 120. 130.
I see black smoke coming out of your.
Oh yeah, that's great. Yeah, so the flush pump is working. So we do have the fluid in our system. It's rising still. I'm at 140 right now. So, and I think I want to fill it up a little bit more because my temperature too, that's the exit temperature, is still fairly low. So we can have it sometimes up to 40 to 60 when the samples are good quality.
No problem.
It's at 155. It's not like super high, but I also feel like we're running out of time to. Yeah.
On your output now.
Yeah, that's starting to get really good. So I'm at 155. Okay, I'm starting piston 2. Yeah, it's gonna be, the sample is moving.
Realization that we were going to be looking at so much hydrothermal. The dates fall back originally. We didn't find anything.
Oops.
This requires a lot of patience. Yeah, but this is clearly how to make the most of it. Yeah. So it could be a place pages. John, what is that skeptical look? Is that a skeptical look? I wouldn't know this, but it's well, then what happens to all the heat source dwindles? What happens to all the prototype, all the alpha matrix? Realistically, the heat source dwindles. Especially it's going to alter basically, so there's not going to be really much synchronization. So you already look around.
No, that's fine. Possibly today it's going to be a geology dive. Most likely today. I'm just not quite sure if how it changed. I'm not sure when we're going to end. I think so.
The problem with Lost City, it's an N of 1. It seems to me it's kind of like a lot of things happening in a very specific way. You know, like I think that in that group we have seen a lot of those things that create velocity, but not, you know, in the correct way to develop velocity. Tomorrow, what was the what was the hydrogen in any of this stuff from the seafloor at Newmark?
What do you mean from the seafloor, from the vents?
Did you take any from any of the bags or anything?
Yeah, up to 3 millimolar.
Oh, so it's still pretty high.
Yeah.
We got close a couple times.
Okay, I finally managed to actually get the sampler started. It took me a little bit longer than I thought.
Anybody have any coffee cups that need to go upstairs? No, that's good.
I'm correct that this is sample 23, right? Yes, the piston 2. Yeah, thank you. Okay, I'm filling up the first piston here. It's almost done, and then I'm taking another piston, and then 2 bags, and then the gas tight, and then more piston and bags. Spreading the love a little bit to different type of samplers we have on board. Okay, that sample stopped. The next one will be piston 4.
Right. That's sensitive eyes.
Okay. Can you see anything on the exit one? I don't. It's hard to see. It's so dark up there. But that's definitely from the flash pan. But yeah, maybe sometimes I think it covers. The color changes a little bit. So maybe that's still like smoke there. So we're halfway through this piston, so I'm filling them up with 500 milliliters of water, and the bag typically have 350, and my gas tight has between 130 and 140 milliliters. However, the gas tights, they're evacuated, so we have a vacuum in them, so filling them up actually takes only like a second or even less. I typically leave the bottle open for a few seconds just to make sure that the fluid really arrived in there before we close it up again. And once we close it up, it's sealed in a gas tight manner, so it has a stamp tip that is golden, and that gold seals everything off in a way that we don't actually lose any gas, even though with the outside pressure changes and the volume of the gas getting more, with just less pressure from the different seafloor and being on board ship, so we know that we still have a sample and we haven't lost any single fraction, so no degassing on the way up. Okay, that one also finished. So the next one is going to be a bag. Bag 16. So I filled up 2 pistons now. Now we are moving the sampler position to the back samplers. So I'm gonna fill up 2 of those, and then I'm taking a gas type. Do you happen to have written down the time from the P4? You missed it.
Yeah, 7:26.
7:26. Thank you.
And to restart this one?
And not quite yet. It's still moving the sampler to the correct position. Now it arrived. I have to tell it how much volume I want. So it's 350. Okay, it's starting now.
See flow out of this.
Can you see? Oh, that's good. Oh, yeah, now I see it too. Yes. Yeah, oh, that's great. That's a good sign. So,
Do we have some time here still?
yes, you have half an hour left
Okay. For the dive?
for the dive.
Oh, for the sample?
Probably half an hour. do you want to sample something
No,
else?
no.
Yeah.
Because it was just to play with the camera around.
You can actually. So yeah, I don't need the view. It's okay if you want to play around.
Okay, perfect.
Yeah. Oh, yeah, our samplers, they don't stay at the pressure that they were sampled at depth. So when we are coming up, the volume gets bigger or it's expanding with the less pressure outside. So I just have like a very high pressure in the sampler itself, and we are hooking it up at the high vacuum line. So the sample never really gets out to the atmosphere. So we have our line here is also pumped down, has a vacuum as well. So we just hook up the sampler on it, and that's how we get the the gas out of it. We couldn't open it just like out by not having it on the vacuum line. So because it was like the pressure is so high in the bottle, it wouldn't be safe. Okay, so this one is done. The next one is back 17. No, it's still moving. Needs quite a bit of patience here. Now it arrived. There was only one position, still took a little bit of time. Gonna pump 350 mils again. Okay, I'm starting. Yeah, but the white with the white background, yeah. Okay, this one is finished. Okay, the next thing I want to do is taking a gas type that is connected to the system, and the last one that is still empty is the one that is mounted aft, so that would be valve 6 port.
Yeah, is that 5?
Yeah, that's at 5 on your diagram, yes.
Yeah, so
And I think that's the one also where we have to adjust, valve 6 port 6. And I'm almost ready. Okay, now I'm ready. The pump is running. Temperatures, okay, yeah.
Which one are we doing? Sorry.
Uh, yes, valve port 6. And it will be the exactly.
Okay. We're
Yeah,
ready to go?
I'm ready. Yes.
Okay.
Oh yeah, looks good. And retract.
Retract.
Yeah.
Retract it.
Good. Thanks.
here for you tomorrow.
Hmm.
So we're here for you.
Yeah, I have them all. I need to log it. Just a second, and then we go on. So that was 27, and then the living extension. Yeah, you already got it. The gas type 12.
Oh man, that's I hit the wrong button. That's gonna make it all wonky. Sorry about that. Yeah. I like these. You like that?
Do you have more than half this now?
Psychedelic shrimp. Yeah.
40, thank you.
Yeah, that was got a little excited on my decrease there, so I hit the white balance on that.
Oh, the flush pump went out when we took the gas type.
Stiff wrist. Okay.
So next one will be a piston again. Yes, correct. Piston 6.
They're fast, yeah.
Okay. So are we pumping 500 milliliters for this one? Okay, starting piston 6. Yeah. Yeah, I think it's coming out. Yeah, there were the white. Yes, yeah. Our temperature is dropping a bit, but the temperature dropped a bit to 120. But I don't know if let's try that. Yeah, I just finished the sample, so if we try to get a hotter temperature again for the last 3 bottles, and I'm writing down my notes in the meanwhile. Still 113. $120. Yeah, it's still 120.
Still 120. Alright.
Yeah. It's very, very slightly. It's now it's 120.7, slightly.
What now?
107.
Keep breaking it.
Yeah.
Pretty clogged up too.
Yeah, that might be also a reason that I'm actually not measuring the actual temperature anymore.
It's pretty solid.
Yeah, it's
Yeah.
on there, it grew on there.
Oh,
It's crazy.
yeah. So precipitated, huh? I don't know if we if we still have access to fluid like this. Let's see what's happening. Well, it went down to 17 degrees, so it still must be reading. Yeah. Now it's rising again. $100, 110, 120, 130. Yeah. 140.
40.
150.
Okay, alright.
We got distracted.
Focus on this game.
155. Okay. Okay, I'm back to 160, so I'm starting setup for the next sample. Yeah. Which is it now? Piston. That's piston 7. Piston 7. The next one. It's close. Okay, starting piston 7. 29, sample 29.
Yep.
Yeah. Yeah, that's good. Yeah, it's 160.5, so it's stable. Almost 170 degrees now. I have 2 empty bags, and I also want to take 2 Niskins, so it's like, is it very sharp, 5 o'clock? Better I'm doing. Up to me, okay. Well then. Okay, this one is filled up also. Here we had a max temperature of 177, was actually a little bit hotter than before, that's great. Next one is bag 19. It's moving. Okay. Okay, starting 19. Still have a few minutes. We're now at 180 degrees, but it could also be because of it kind of gets clogged up the temperature. It has its own little environment. I'm not quite sure. What a surprise, huh? One 183, but I'm not quite sure how the temperature probe is currently behaving. With the high temperature one from the ROV group, we have 300 at the same spot. Okay, this sample is done. I do one more. It totally fits into the schedule. So the last one will be back 20. Okay, starting the last one. Yeah, that's great. And the temperature is not 196, but I'm wondering if it's just clocked off. Yeah.
Tomorrow?
Yep.
When are we supposed to depart?
No, but so I'm filling up this last pack. It has like 2 more minutes, and then we want to take 2 skins in the smoke while we are rising up.
Okay. Hey, Tomar, do you know how far we are from the AUV recovery site?
Not yet, but we'll check.
Okay.
I think it's very far at all.
Well,
I think it's in the dive plan, the distance. I don't think it was far.
yeah, the various plans are.
Oh, okay. Okay.
Oh, yeah, it's a different word.
No, but I think that the actual distance was written down between here and the AUV pickups.
So we should be able to track that down.
Yeah.
SPL parts. Up there. That's nice. Very nice. Sad to see for 2.5, 3 hours. I think I flew 15 meters. Great. So I just sample the one that
Okay, my sample is done, so we can store the wand. Yeah.
How's that been working tonight?
Very good. Yes. I didn't have any issues at all. Yeah.
All right, I think we sorted some things out.
Oh, that's great. Yeah.
And I'm hoping your other gas lights came out good.
I hope so. We will find out.
A little weird.
Yeah, I didn't know. It just looked so short. Everything, but maybe it was just the angle we were looking at. I'm not sure.
Open up the iris. I did.
Yeah, don't click that one too much. Use your iris more than anything. So open it all the way. Yep.
So we're done with all the fluid sampling. We are just storing the wand now, and then we will rise a few meters up, close 2 Niskin bottles, and that will be the end of our dive.
Faster than you. It is just 145 channel. You're gonna be taking a Niskin, right?
Yeah.
In this spot, or you're gonna want them to get up and over it?
Up and over. Yeah.
You might as well just back out. You're not gonna be taking the Niskin in this spot right here. So if you back out, you can see what he's doing.
Yeah, we can I don't know, 5-7 meters above, something like this.
Yeah, just went back. Okay. Don't forget it to your right.
And probably to your
Yeah, you can do whatever you need to the cameras and all that. That's fine, right? Okay, there you go.
Yeah, don't open bottle boxes. I think it's full of chimney and shrimp. Monica's gonna be busy.
Sorry?
So you're gonna be busy.
With what?
On the maybe Monica won't be busy.
I will be.
Tomorrow, how much time do you think you need to finish?
Now I'm done. So we are storing the wand and then moving up like 5-7-ish meters, and then close the 2 Niskins in the smoke, and that's it.
Okay. Maybe we can do them at different heights.
Yeah, however, up to you what you prefer. Yeah.
What do we have left? We have,
We have 2 left, and
which ones are they?
there are 3, which is good for sure, and 5.
Can I use one of the?
5 is a 2 liter one. Yeah, they should,
That's a
they
small
should be
one.
fired at the same
So-
time.
okay. Any preference how high up, or you want to have a look when we are rising?
Sort
Or?
of, well, I think roughly 10 meters.
Okay.
Well, it was nice enough to put it into a vial. And they just blow that conversation. Goes with it. And then,
those are all
probably
end up in there.
Can't carry it back.
Between the porch.
But Sean might do it as well.
Yeah, okay.
Yeah. I don't have to go to customs.
Which one do you want to do?
I saw fire.
Which Niskin?
The Niskins will be 3 and 5, and Joe would like to have it about 10 meters above seafloor, approximately, in the smoke.
He wants to get there an hour early
Oh, yeah, 3
because of a 20-minute. No, that's not for me.
So you get to the
Yeah, there was something weird, like they wanted to get there and.
It's basically, I think they're on. They
really difficult to see
They
once they
want
get to the
every
smoke.
possible way of knowing where it is, and they don't want to take any chances. They don't really admit that.
Yeah.
So they can not come in and say, oh, it's going to cost 20 minutes to get a satellite. Like, so I want us to be there an hour early. It's going to cost us 20 minutes to get a satellite.
What do you feel about these samples?
I think good, yeah. If the temperature is just different to say, like how hot it
And,
is.
are we still
Like
talking
takes,
about
and
some
I'm
tools
not
that
quite
can
sure how
roughly
air pressure.
Yeah,
We have
roughly
300 on yours,
Yeah.
and they go for like 160 maybe on ours.
Who we are.
I mean, you guys, it seems like everyone's got to retool, renew stuff and retool.
Yeah, as long as y'all have all your stuff ready to go.
That's what I just did, take on my stuff.
You said about 10 meters up?
About 10, yeah.
Yeah, just get the Niskins in that smoke there, like come up and put those Niskins in that cloud as much as you can, not where you can't see.
But at least you get 10 meters up.
Yeah, keep coming up.
I mean, we can say that, but I think my side more is something.
Yeah, it's any week.
It's crazy to try to turn things around that fast. It's driving you crazy.
Try to go something.
checking the time.
Mm-hmm.
Because I have my timing.
Local?
Set it, yeah.
Yeah.
Local, so it's 8:09. Is that one there?
That one is correct, yeah, that's the UTC time.
Manufacturer must be had the loader rebuilt by, but they must not have it correctly. So the electric motor inside there with all the windings and everything like that, the main input of electricity from the umbilical, those are two separate circuits. And somehow those circuits are crossed on, and that's the only spot those two are.
Yeah.
And they had to rewind it, so they would have undone that potting, they would have rewound.
By potting, you're talking about a seal against seawater invasion and fluid escape.
No, it's really just a seal between cross-hot wood. That's all that seal, which is supposed to be low pressure, and you should not see very much pressure. Both sides should be ambient, 6 to 7. Exactly. So it should be seen. It's not proper like potting like you would for a cake. That should keep out elements. But it's still trying to
You got something right off the start right here. And it's all right. And that's your port. Just the starter on the back. Yeah. There's nothing that's.
What's up?
Y'all lost in the cloud.
You're still right above it. Look at your map. Yeah. And you are.
Yeah, we don't have everything.
Yes. Do you hydraulics picture? What? Hydraulics picture?
Now I can see. Still definitely the same. Let's go.
You are a rock as well.
Yeah.
That means there's legitimately a full chimney inside.
Yeah, probably very happy.
Well, we got some more.
And there's another one.
Extra
Got a whole
one.
extra one for everybody.
I think there's more on the quivers.
There's like a whole bunch on there.
Mhm.
They'll took something out.
Yep. It's heavy too.
Like a steel plow. It's really heavy. Give yourself some. There you go.
There we go. Alright. So
Victoria Bone.
it's come up 10 meters, but you kind of
Just keep paying them, Chris. Make it hard for them to come down.
let's get like 20. It's coming up. Alright, 6.5. About that. There you go.
It's amazing, huh?
I crossed that one to your right.
Wow.
Okay. Above everything, I think. Alright.
Go test, get
3 and 5. Happy here?
Yeah, that's good, right, Joe? Happy?
Yes, happy. Perhaps too happy.
Alright, yeah.
How high are we in what?
7 meters, sorry.
about fluctuating between 7 and 10.
Okay.
Depends on what peak it's using the beam off of.
Sorry. Trying to find down in that high. Haha. Got it. That was awesome.
Alright, let's start coming up.
Great, thanks.
Which number of sample it is?
That was 32.
32.
Yeah, and then Niskin 3 and 5.
Yeah, perfect. Noted.
Noted.
Combine that load and try to grab it again. Yep. Take a look to your right with this knife.
All right, so that was it from our side. This is the end of the seafloor time. So we are coming up now. It's going to take about 3 hours until the ROV is back on deck. And in the meantime, like all the scientists are getting ready to prepare for all the samples we're going to get. So like, for example, I'll go and calibrate our gas chromatograph so that I'll be able to run some of the gas samples. The biologists and the geologists are getting all their description materials ready with cameras and sample numbers and microscopes, and the fluid chemists are going to label their bottles so that they can take all the subsamples. So it's typically very busy in the time during the ascent and before we know that the ROV is coming back on deck.
The port fell away in.
Can you string on the ascent?
I don't mind.
What? Excited about being out of
Solid amount of samples.
Only if they want to distribute.
Oh, we can do that.
And the power to the HFS can be turned off whenever you're ready.
Gotcha.
You mind if I try to grab a worm, dude?
Yeah, I'm still facing south. I was just talking about that. So.
Chris, did you grab this? What do you do? Bites.
Oh, that reach?
Yeah.
Because they were all remembered. Yeah. And I found that we put the chimney, was all pretty little, they kind of like get a bunch of stuff. Yeah.
I got a pretty good view on that.
That was not the plan. How did you do your job? How do you want to stay with that? Yeah. Until we arrive. Yep.
So come down.
I will stay with my
Because you met again.
last night for yeah.
Let's put it right about there.
Bring it out some for you.
Yeah, how much more? Either rotate it out or lift it up.
Good, and now we'll get on deck.
I guess I'm actually just also
Handle.
Shoulder right. Tuck it in.
some moment and the air.
Oh, is that true?
The air parts can
That should be good right there. Thanks,
Well,
But I feel like I you that.
yeah.
I think the more controlling things in the effective
We should be safe when we can drop.
I'm still asking, but I'm also interested.